We start. Yeah. Oh, yeah. So good morning for everyone, and I'm sorry for the short delay, but well, always we need to set up the things and the microphones and everything. So good morning, and welcome to this event, Enzyme Solution for Green and Consumer Product. That so it's a final event that brings together the research that we for European project make in the last 4 years, and then today we are partner and coordinator for the 4 project. Also, people from interested in enzyme technology, in consumer product, in greener product, in sustainability, and so on. So today, what you will hear is the result of this 4 project, innovation that we achieve in this project, and also we will have round tables. So we're experts in the field. So we are going to discuss, or they are going to discuss about the topic that are relevant to the project. First, I want to thank the Fundación Telefónica because they offer the possibility to make the event in this nice place. Also, thank you for you, those that you are here in person, and also for those that are online. Thank you very much for attending. Also, thanks for the different partners of the project, and also for the person that will constitute the round table. And before we start the main, I think, before we start the event and the agenda, which is here, so we are delighted to have or to welcome 2 special guests. For us, it's an honor, a privilege to have. First, I will introduce Isabel Diaz. So we work together in the same institute, in the Institute of Catalysis and Petroleochemistry. But then now she's Deputy Vice President for International Cooperation at the Spanish National Center for Research Council, the CSIC, in Spain. Yes, here is the photo, but you see this. It looks she didn't change. So it's because sometimes we put photos that are really. So it's So just in summary, I want to introduce Isabel. It's really a pleasure to have her here today, because today is a really busy day for her. So we are thanks. So she holds a PhD in chemistry. Then she makes a lot of research in Sweden, Japan, in USA, and so on. And then at the CSIC, she led the science diplomacy effort, and also this international cooperation, and also the international research infrastructure at the office. So thanks, Isabel, for being here. And then we are also pleased to welcome Colombia. It's also the same, the photo. It's, you can recognize really well. And then she's our project officer. Is the project officer for our project, FuturEnzyme, that you will listen later on. And then she's a project advisor at the European Research Executive Agency, the REA. And she has a strong background in communication campaign and European affairs. And since 2011, she has been working in European research and innovation program in projects related to biodiversity and climate change. So Isabel and Colombia are going to give some words. So it's a great pleasure to have you both here. And then, without extending more, so I officially open the event. And I forgot to say thanks Gro, the coordinator of OXIPRO, Aurelio, the coordinator of RadicalZ, and Carolina. And Carolina is coordinator also of EnXylaScope project. And just to be here and to organize this event together. So so that's all. Isabel, the floor is yours. So thank you.
Well, thank you very much, Manolo. I don't know if you call him Manuel Ferrer or Manolo, but I know him for 25 years. So today, my job is basically a pleasure. because being here today with the colleagues that I know for a long time, makes my day happier. Anyway, thank you very much, all of you, for coming to Madrid. It's a beautiful day. You're in downtown Madrid. Please walk around. Use the time. Now the sunset is later, so you can walk to the Retiro and enjoy the city. Because, we also have CSIC, we have the House of Science. I don't know if you remember the TV show Money Heist. Well, the headquarters of CSIC is the Money Heist. La casa del banco. La casa de papel. No, how did they call it in English? The bank, yeah. Well, the house, the main building, is the CSIC headquarters. We are the largest research performing organization in Spain. I mean, we really are the scientists of the government in Spain. We're not a university, but even though we say council, we're not a funding agency either. We are a research performing organization. More than 16,000 people. Actually 16,800 this year. A lot of people. 124 research institutes. We know one, but there are 124 in every topic that you can think of, except of research in law, basically. We also have an office in Brussels, the delegation in Brussels, that helps us doing the so-called lobby. We love that word. We don't really know what it is. But anyway, one thing we do well is getting funds in Europe. We are number 3. We are in the top 5 of performers of science that gathers money from Europe. Why? Because we need it. Only half of our budget comes from the central government. The rest comes from the overheads of the projects. So we do have to go and look for funding. And that's why making communities like this is super relevant. Because science cannot be done alone, and especially reading what you achieved and you have done in these 4 projects, the way you approach science and greener solutions to society, it for sure needs teams, needs people, because you're doing science for people, right? You're aiming to reach the market, but when one reaches the market, what you actually reach is individuals to make our life better. The planet a little bit greener. I like this greener thing, not greener, right? Because oil is also green, right? When people say, let's do natural things. Well, petroleum is green, is natural, yeah? So your greener part is particularly interesting. So just wanted to welcome you to this and just make you feel like you are part of something big, because I'm sure you are aware of it, but when we are in our atom and in our enzyme, we lose the perspective. So I'm just coming here to tell you, please don't lose the perspective, step back, be proud of yourselves, be happy about your work, and just use that to motivate you to get more. We will be there to help you. We have 2 or 3 small things that allows us to move people from one place to another, small mobility calls. We make a big effort in sustainable development with low and middle income countries. We have special calls with Latin America, Africa, Asia. We also work strongly in science diplomacy to place science at the speech of our politicians. Why Donald Trump knows why he wants to buy Greenland? Well, because scientists told him to do so. So that's what we want to do. We want to be a tool for our diplomats and our politicians to do or make a better world. And finally, we are committed to gender equity. CSIC got the gender equity champions from European Union this year, and we are working hard on having safe environments for researchers. So with that, I just want to thank you for inviting me today and enjoy the conference. Thank you very much.
So I can start. Yeah, thank you very much, Isabel, for your kind words. And I confirm I have CSIC in many of my projects, and it's a pleasure. It's a pleasure. So buenos dias, everybody. Good morning. I'm very honored, and it's a bit with emotion that I'm here today, because it has been a journey together with Manuel Ferrer. I'm the project advisor of the project FuturEnzyme, one of the 4 projects from this cluster. And 4 years. That's came quite fast. A lot of things around us in the world, but I liked Manolo because you have been always directed to the point, the description of action, as we say in our jargon, and really committing with all the partners of FuturEnzyme and the EnXylaScope, RadicalZ and OXIPRO to work together. I must say, as a project officer now for 14 years, it's quite rare, and I really want to insist on that, to really have, after the end of the project, some tangible results. And this is the case for FuturEnzyme and for the other projects, and I'm very happy about that. And I wanted also, I had a reflection about how did it work, thanks, of course, to Manuel Ferrer and from CSIC, but and the partners. But I also think about a very good combination in the project with research partners, academia, and on the other side, companies. But companies that really were there to cooperate and that see really an interest to cooperate together with research. And I must say, and you mentioned Trump, okay, but we live in a complex world, not only because of Trump, but also because of China, because of all things. And we are a bit criticized in Brussels at the European level for having excellent researchers all around Europe. But sometimes we have this path from research to innovation, and in this little part we are, it's a fundamental, not little, but little path, we are not the best. And I must say that here is the contrary. Today I can say that we made perfectly the path between research and innovation, between enzymes and products. And yesterday I attended the General Assembly, the last, unfortunately, General Assembly of FuturEnzyme, and I was very happy to see that we were not just talking about, okay, we identified enzymes, we selected enzymes, we'll see how it works. No, we were talking about really the key lead candidates that they were going to be taken by the companies. We had Scholler, we had Henkel, we had Evonik in the project, and that it will become a tangible product. And for me, this is a success. And so this aspect that I wanted to highlight, the first aspect about this move from research to innovation. But the second aspect, and that's also very close to my heart, is the fact that we are not talking in general. We are talking about very clear products, cosmetics, detergents, textile. This is in everyday life of citizens here all around, not only researchers. So at the end, with projects like the 4 projects we are going to hear more about today, we have an impact on society. And this is super important, because we have seen also in those past 4 years some changes in Europe, European Parliament's elections. I mean, maybe some shifts of citizens that have some doubt about the EU are voting maybe not exactly with constructive politicians or putting not very constructive politicians in the European Parliament or in other member states. So I think that if we manage to really talk about the impact of projects like this, everybody will understand the importance of the European Union and how we can work together and be strong together. So, yeah, I just wanted to end with that and to say that for me, this is the perfect recipe of projects that work and that have tangible aspects. I want now to say, just before giving the floor to all the coordinators that did a fantastic job, to talk about my colleague, Mafalda Quintas. So she cannot be here with us today, but she will be joining online in the afternoon. And my colleague from DG Research, I work at the European Research Executive Agency, so I monitor projects. But my colleague, Mafalda, she's working on everything regarding the calls, what we call in a little jargon, the work programs, and she will shape a little bit what's going on for the future, what's the policy framework. I think also something that you did very well here in this cluster, to shape the research data and the research products next to the EU regulation, because we have regulation in the EU, which is good for the environment, by the way. And she will say more about that this afternoon, so I'm very much looking forward. And I'm very happy to be here all day to listen to the key results of the projects. Yep.
Hello all, I'm Nerea. I'm from Science-C, the science communication agency that is part of the RadicalZ project. But most importantly, I'm here as your MC, so I will be introducing the different speakers and moderators for today's event. And honestly, I've had the pleasure and the honor to work with all these 4 projects for the past 4 years. We've been collaborating in the project since the very beginning, and it's been a beautiful journey which I hope gets to shine and to show the wonderful results that these brilliant researchers and teams have achieved. Anyway, I don't want to take much of your time, so let's kick it off with the first in line, which is the EnXylaScope team, and we will welcome to the stage Eva Nordberg and Arnaud Marcelin. Eva Nordberg-Carlson is a professor in biotechnology at Lund University and the master contact from EIT Food and Lund University. Her research focuses on enzymatic and microbial technologies for biorefinery applications of agricultural and marine resources, with a strong focus on carbohydrate conversions, especially related to increased side stream utilization. And she's currently part of several European projects. And on the other hand, Arnaud Marcelin is an organic chemist specializing in biocatalysis. He began his career at Arkema in France, developing enzymatic processes, and then he earned his PhD in carbohydrate chemistry at CERMAP, also in France, and subsequently joined the CEPIC to establish a biocatalysis activity. So may they take the place on the stage. Thank you.
Thank you very much for your kind introduction. I don't know if you're going to wait or if you're standing here along. So we are here representing the EnXylaScope project. Very happy to be here. So the project's name is Unleashing Silence Potential with Enzymes for a Scope of Consumer Products. And I am sort of the academic representative, and Arnaud here is one of the company representatives. So we are, in all, we are 13 partners in the project, as you can see here. And if we move to the next slide, I don't know who has the control, or I have the control. Great. Then you can see that Xylan is really the thing here. Do I have something? Yeah, all right. That's how it worked. Xylan is a hemicellulose. It's one of the most abundant biopolymers on Earth, but surprisingly, it's still underutilized. And that might be because of the fact that it's substituted in different ways. So a little bit dependent on which resource you are using, it can be substituted with different types of side groups, like glucuronopyranosyls or arabinofuranosyls or ferulic acids, and that is something that we want to utilize in this project. So a little bit opposing what people are normally doing, we only want to debranch. We don't want to break down the polymer with these main chain cutting enzymes. We want to just take off side groups in order to change the functional properties. And that's where you have a little bit of lack of enzymes currently. So surprisingly, despite the fact that people have worked with xylans for a really long time, the debranching enzymes are not that much looked at, and specifically not the ones that are acting on the polymeric substrate. So if they act on oligomers, that's not what we want. We want them to act on the polymers. And I'm here in this first part. So you can see that Lund University is in the first box where you have the discovery of these enzymes. So we are together with Sintef, Delft, and Metgen. And then in the next box, you have different companies, like, for instance, Selignis. We have Oscar sitting here in the audience as a member of the company that is actually using the enzymes to do these products that are then going further to the companies which are testing them in the applications. And we are also happy, we had a meeting yesterday, so we saw that on the sustainability point of view, Heiko, who is sitting here, was just mentioning that it could actually be advantageous to use the enzymes from a sustainability point of view. because the purification procedure is easier. So that's good from that perspective. It's 4 main groups that we are looking at. It's glucuronidases, arbenofuranosidases, and these esterases that can remove acetyl and ferrolate from the resources. So how did we go about? There is an enormous amount of genetic information available in databases, and we also had metagenomes available at the different partners. So we were selecting, in principle, sequences from these different families where you could see that these enzymes are actually classified in databases, so you can find sequences and then search for more that are a bit related when it comes to the different sequence information. And then we clone them, express them, and purify and characterize them. So what are then the main bottlenecks? So we used E. coli as our production system, and that part was considered because our upscaling partner, Metgen from Finland, they preferred that system when it comes to the upscaling. So then we used this system in order to generate something that was easy to produce, where you can see a rather big amount of the enzyme. But what we also looked at was, is it soluble? After you have produced it, is it soluble and is it stable? So we, in principle, we purified a lot of the candidates that look promising in order to be able to look at those perspectives. So solubility after 24 hours and after purification was one way of assessing the enzyme. Another thing that was of importance was, of course, the activity. And that is not as easy when you're looking at polymeric substrates. So there are plenty of screenings possible if you are looking at small molecules, but the high throughput alternatives are a little bit more tricky when it comes to looking at the polymers. So simply we were trying to find methods, for instance, with chromatography, where you could see that the molecule that we wanted to remove was actually visible, so that you had that as a target. So this was the main way of looking at these enzymes, and by doing that and looking at solubility and stability combined with the specificity, we found actually candidates of all these 4 groups that were good in terms of storage, stability, and production, and had the function that was desired. So from that perspective, we have now listed the ones that were most favorable that were used by the companies in order to get the trials out to find the xylan that had the function altered, so that this would be of interest for these, for instance, cosmetic applications that were tried at CEPIC. And with that, I will just say thank you for me and leave the word to Arnaud.
So thank you very much, Eva, for your kind presentations.
Yeah.
And hello, everyone. So I will introduce what CEPIC job was in this project, and also why we joined this project, which is quite interesting for us. But first of all, who are we? So CEPIC is part of the Air Liquide group, which is a huge French company. So the main activity of Air Liquide is more gas, like oxygen, nitrogen, but we still are part of this company. We have quite different molecules that we are producing. So at CEPIC, we are mainly based in France. We are near to 9,000 people. The main factory in Castres was in Castres, in France, where we have also RNA research. We also have other facility in France, in Pau and also in Brittany. We also have facility in China and another one in US. But what is very important at CEPIC is that we have a technical center all around the world that can allow us to supply our product and also to have the need of our client. So basically, we are in different markets, but our main market is about beauty care and cosmetics. We also pharmaceutical, nutraceutical, and other markets. But the main products that we are producing are thickeners, polymers, and also surfactants. So all these products are mainly produced in the facilities that we have in Castres, where I come from. So why do we join EnXylaScopes? So I think Eva well introduced the project, but I like to keep this xylene structure to show how it is complex. What is very important is that carbohydrates come from wood and are very poorly evaluated. They have very complex saccharidic structures and with a lot of ornaments. So the objective of the EnXylaScope project is to discover, evaluate, and scale up new enzymes that have the ability to deconstruct this saccharidic ornaments, which is quite a challenging project. But we will also go much more far away for that, because we also like, thanks to these enzymes productions, modify some xylene and find application to the xylene for potential markets. So this well match with the CEPIC strategy of development of ingredients, because we look like ingredient with high performance, and I think you are convincing, but thanks to enzymes and thanks to their selectivity, we can have some specific molecules that will have this performance. This is what we're looking for in this kind of project. The other important stuff that we have is the sustainability and eco-design. And as you know, biocatalysis process had quite low carbon impact, which is quite interesting. But also, as the xylene are from wood, it's a raw material that could be traceability and also renewable. So this is very interesting for our strategics to use this kind of raw materials. There is also naturalness, because it is very important in cosmetics for our customer to know that it's from nature, it's biosourced. And also, we also have to manage all the safety and compliance that is requested to put a product in the market. So, like that, I think that EnXylaScopes match quite well with CEPIC strategy to develop new ingredients. So what we do in such kind of project, because we look for some application for that. So here you have all the partners that Eva already kindly introduced. We are at the application stage with Kerry, that Laura is here, which is in charge of using the enzymes application. We are in charge to find, thanks to the xylenes, the new xylene generated for this enzymatic modifications, new performance for the cosmetic market. What does that mean? We evaluate some xylene property in formulation and more precisely in emulsions to see if we can get the stability and if we can also benchmark the products that are already in the market. We also was in charge of the toxicological assessment of this product in order to test it by consumer and to have a feedback from the products. So what is an emulsion? So I'm quite sorry because I had a video, but it seems to didn't work. Emulsion are quite basic. It's just mixing water with oil and surfactants. Then you mix this, and then you get an emulsion. And as you can see, you can have many applications in cosmetics, such as skincare, makeup, and so on, that could use this kind of molecules. So in this way, we look for performance in this way with xylene materials. Oh, the video is working. Oh no, it will just have the picture, sorry. Here you have what looks like an emulsion. But it's quite easy to make an emulsion, but what is a challenge is to have an emulsion with stability. So for that, we have to check the stability during at least 1 year at room temperature, but also for 3 months at 45 degrees, which is quite challenging because it required a quite large amount of time, so that we have to manage to check that there is no instability, as you can see right here. So this is quite a long time, and during these times we study pH, viscosity, and also the visual and microscopical aspect of the formula. So we compare all this performance with a benchmark to know if you are better or not than what already exists in the market. So it's exactly the job that we do in the EnXylaScope project. So we get some samples from Selignis, which use the enzyme producing during the project to modify the xylan. And thanks to that, we screened a lot of different combinations of enzymes to generate this interesting xylan to find some emulsion properties. So just here you have some emulsions that we have thanks to the xylan that was produced in the project. And we evaluate all this performance in the formula. And we had quite some interesting results, but of course I cannot say much more about it because we like to patent it to move forward and to maybe put a product in the market in the future. So I hope that this showed to you one example of what we can do thanks to biocatalysis and also thanks to the production of the enzymes. So I will thank you very much for your kind attention. And if you have any question, I could be here to talk.
Yeah. Yeah, for sure. So we leave the questions for later.
So.
Thank you.
Thanks. After the presentations of all the different projects, there will be an open Q&A with all the speakers. So maybe if you have any questions, save them for later. Right now, we're moving to our next project in line, which is RadicalZ. And I'm honored to introduce my project coordinator. Aurelio Hidalgo is a specialist in enzyme discovery and optimization for biotechnological applications. He did his undergraduate and graduate studies at the University of the Basque Country and was a postdoc at the ICP-CSIC in Madrid and the University of Greifswald. He then joined the Universidad Autónoma de Madrid as a Ramón y Cajal Fellow, and he's currently the Associate Professor of Microbiology. He also leads the High Throughput Discovery Lab at the Centro de Biología Molecular Severo Ochoa here in Madrid. And he has led several national and EU-funded projects dealing with different aspects of enzyme discovery and engineering. He currently participates in the European projects MetaExplore and Extreme, and he coordinates the European projects Blue Tools and the one that we presented here, RadicalZ. So, Aurelio, welcome to the stage.
Gracias. Okay, thank you. Good morning, everyone. So I've been introduced, so I don't need to introduce myself. What I need is to make emphasis, there we go, on rapid. So this project is about accelerating the time that it takes for enzymes to go from the lab into the market and the products we develop with these enzymes. So with that in mind, I am going to give you a few facts and figures about the project. Then I will introduce the core concept of the project, elaborate a little bit more on certain innovations all across the pipeline between the lab and the market, and then present 2 case studies of molecules that we synthesized. So, as you know, all 4 projects were granted under the FNR 16 topic to run for 4 years with 6 million euros. We, I think, are a really good example of what Colombe mentioned. We have 6 academic partners and 6 industrial partners, and they worked in tandem. These academic partners and companies are spread all over Europe in 8 different countries. At the core of the project is the enzyme. So discovering and optimizing enzymes fast. This is usually the slowest process and also the most, expensive part of the pipeline. Once you have the enzymes, you can, of course, use them as ingredients or to synthesize ingredients for greener consumer products. There we go. And a core concept of this project is that for every reaction, we always start from a molecule that comes from a waste stream. Right? We didn't start from any molecules that come from crude oil. Finding enzymes fast, optimizing enzymes fast. This is done in this project by using 2 core technologies. One is machine learning, and the other one is microfluidics. Also, using microfluidics, we've been able to develop new formulations for the enzymes and the ingredients in the consumer products. And finally, another core concept of the RadicalZ project is we measure. We measure the processes we're developing, we measure the sustainability as it's developing in order to feed back as early as possible to design. So, as I mentioned, this is, I think, the longest and the hardest part of the pipeline, discovering and optimizing enzymes. So when you're trying to optimize an enzyme, you replace one amino acid with another, right? So for every position in a protein, you have 20 possibilities. And then if you have L amino acids, you have 20 to the L possibilities, and you need to navigate that. And navigating that is like moving through this complex mountain landscape. You need to get from point number 1 to point number 2. But how do you do that? Well, you do that with trained help. And this is what we did. So we used machine learning, artificial intelligence as a help to engineer proteins fast. So here the team from Bioproduct developed new protein representations. On the left, you have the non-bioinformatician translation. That's the one that I use. And they also developed and trained a protein language model with unlabeled and labeled data, so public and proprietary data. And here, I think the strength of Bioproduct as a partner was that they had this proprietary, there you go, massive structural alignments and pathogenicity predictor that has allowed them to train this protein language model. And you can see in the bottom there that with every layer of information that you train the language model with, it gets better and better and better until it's best in class if you compare it with other existing AI tools out there. And now that is actually a product, a service. It's called 3DM engineering. Now, while they were developing, we were eager to test, so we tested a preliminary version of this predictive tool. And what you can see in purple is the result of 2 years of blood, sweat, and tears, as we said yesterday in the General Assembly, right? This is a conventional protein engineering campaign combining protein rational design, directed evolution, recombination, so on and so forth. With the information from our 2 years of blood, sweat, and tears, they trained the language model, and in a lot shorter time, they came up with the green labeled protein that you can see. And we tested both of them in the final application, and as you can see, depending on the conditions, they have different performances. Sometimes one is better than the other, and sometimes they're comparable. When you have a good starting point, then you run a directed evolution campaign, but sometimes it's all about finding the starting enzyme for a process. And here, Brain used a proprietary database called MedExtra with millions of natural sequences that they process using bioinformatics, and use, again, machine learning to identify where the interesting candidates are. And look where the interesting candidate is. Sometimes nature surprises you and gives you a starting point that is even better than the engineered enzymes by their method. Yeah, so don't rule out nature just yet. Yeah, and that goes actually into scale up and production. All right. Machine learning, bioinformatics, that requires information to begin with, but sometimes you do not have the luxury of information. And then you need to resort to function-based screening or directed evolution combined with very powerful screening. So you actually need to screen one by one every individual in a library, and these libraries are millions of individuals. In order to be able to do that within a reasonable time, we resorted to microfluidics. So microfluidics allows you to manipulate small volumes with precision using these devices that you can see, there we go, here on the screen, or that you can see here in my hand. This is the size of a microfluidic chip, a microfluidic device. With different models of chips, we can do different operations that you would do in the macroscopic lab using large volumes, but we do them in picoliter volumes and at thousands per second. So here are some examples. Here we are forming little water droplets. These are test tubes, which means picoliter volumes, no more plastic. You can add reagents as you would add to a test tube. You can carry out your enzymatic reaction and separate the positive test tubes, the positive droplets, from the negative droplets. So you collect them, and then you recover the gene that codes for the enzyme that was improved for your reaction. If you operate and if you screen using droplet microfluidics, you're orders of magnitude faster, orders of magnitude cheaper, and orders of magnitude greener and more sustainable. We use 5 grams of plastic to screen 1 million individuals. All right, some of the things we did with droplet microfluidics. We developed assays and screening technologies so sensitive that we could start from a single molecule of DNA. We developed real-life assays that measure the polymerization and depolymerization of artificial and natural polymers. And we used them, as you can see in the video, for an accelerated directed evolution campaign in which we also generated large data sets, so large data sets of information. So in total, we developed 5 assays, which is around 10% to 15% of all the total existing droplet assays known up to date. Sometimes enzymes are the ingredients in the consumer products, and sometimes we use them to synthesize the ingredients. When enzymes are the ingredients, they need to be active and be stable in the matrix of the consumer product. Once again, using microfluidics, we were able to generate these capsules that we fill with different ingredients or enzymes, and these capsules are designed to trigger the release of their content when the conditions change. So we can release the ingredients in a consumer product when and where we need them. And we also use droplet microfluidics to multiplex all the additives and conditions and concentrations that you need to test before you put an enzyme into a consumer product, except using picoliter volumes. All right. What about synthesizing ingredients with enzymes? So, as I said before, we made it a core concept of the project to always start from waste. And we addressed 3 types of consumer products, always starting with waste. Here you can see personal care and cosmetics. And here you can see ingredients that we synthesized for laundry detergents and also for nutraceuticals. In total, we developed 26 synthetic methods, enzymatic synthetic methods for ingredients in consumer products, all starting from waste material. But it's not about the number, it's about the diversity of ingredients, the chemical diversity that we were able to synthesize with enzyme. I think this is a testament to the versatility and the power of enzymes for applications, for consumer products. Now, I mentioned that we are also the metrics project. We measure and we make informed decisions. And sometimes you hear blanket statements like, yes, everything is sustainable or a process is green just because it runs in water. Yes, but how green? How efficient? So the University of Zagreb develops kinetic models that have been used to develop our processes. These are these very complex equations with which you can describe the enzymatic reaction extensively. You can use them to predict what will happen under different conditions so that you can actually pick the optimal condition at which your project is going to run, and this informs the scale-up. And how green is the process? Well, in most cases, we will resort to life cycle analysis. We have an example of those, or a couple of examples of those. But when your process is mature enough for life cycle analysis, then maybe it's too late. Maybe it's the end of the project. So here, Sustainable Momentum, Pablo Dominguez, developed a set of, say, simplified equations. The concept is not simple, but the equation is simplified so that everyone can assess the environmental sustainability of the reaction they're developing, so that you can make changes when you're developing the enzyme, when you're assembling the reaction. And you can feed early to project design. So Colombe mentioned that, yes, we work among scientists, but we work for the general public, industrial stakeholders, policymakers. So what's in it for every one of them? So in our project, our output is extremely diverse. So our output, our innovations have been the new enzymes and enzyme variants that we developed, the reactions, the technology that we used to discover, sometimes things that are a little more intangible, and sometimes we have addressed actions to specific stakeholders, starting with industrial and academics and scientists. We have not only organized events. Tomorrow there will be a training school. This Manolo and the rest of the sibling projects co-organized this today, sponsored a conference or gave lectures, but we are actually reaching the market. We have launched, or we have filed for patent applications in the lifetime of the project. This means that there is real interest for uptake from the companies. More than 25 follow-up cooperations, so that these innovations in their beginning will still live on after the life of the project. But we also reached out to the general public. One way to do it is through social media, and here ScienceSeed took the lead, and they have this proprietary tool to analyze interactions and traffic in social media. And with this tool, they were able to target communication so that it was more efficient. And I think it really shows when you look at the metrics that they provided us with. We haven't forgotten about the youngest ones. So we developed educational materials. There is a comic book translated into more than 11 languages. There is a digital escape room for high schoolers. We've participated in the European Night of Researchers and other outreach events. So to finalize, I will zoom in on two molecules that we've synthesized for personal care and cosmetics. So the first one is the raspberry ketone. The raspberry ketone is one of the strongest aroma compounds there is, right? But the natural raspberry ketone runs for several thousands of euros per gram. And you need kilograms of raspberries just to extract a milligram of compound. We proposed a synthesis that starts from wood waste, from birch bark, right? You use the wood for something else, and you use the bark to extract these two compounds. The problem here is how to make that extraction sustainable, or more sustainable. And another problem is that these two precursors, their composition, their abundance oscillates with seasonality. So we actually needed to develop two reactions, one for each precursor. And then from each precursor, you may get one of those two alcohols here. So once again, the University of Zagreb needed to develop two reactions and make sure that the whole thing worked nicely together. Even with a process at a low technology readiness level, preliminary calculations show that we are competitive with the natural product. Why the natural product? So why is this not considered chemical? Because it's synthesized with enzyme and processed with bio-based ethanol. Right? So you can label it as natural, which is strategic for our industrial partner, Analyticon. Now, when we measure how sustainable are we, there's room for improvement. We couldn't benchmark against the chemical because, like I said, we benchmark against the natural product. So what we benchmarked was against the chemoenzymatic approach. Just by going fully enzymatic, you reduce 10% the amount of CO2 that you produce. And just by using renewable materials at the start, you knock down another 60% in CO2 emissions. And when you run life cycle analysis, then you can pinpoint the specific places that you need to improve, like extraction of the precursors or the CO2 emissions from the energy mix. We have this ongoing joke in ESOF where if this project is run in Norway, then our footprint is going to be a lot lower because of the energy mix used there. Yeah? Now, another target compound or family of compounds were antioxidants, right? This is BHT, which comes from crude oil, and it's the antioxidant of choice. And if you want to resort to natural antioxidants, there are not many out there, and they end up giving up a foul taste or a foul aroma. So here, Analyticon and the University of Greifswald co-developed this synthesis. And it's a synthetic reaction. It's a coupling reaction. You see two halves on the screen. Well, doing that with enzymes requires using organic solvent, but this is something we want to try to avoid. So we used an enzyme that excludes water from the active site, right? And in that environment, then you can get or you can reach the desired product. So with some suggestions from Sustainable Momentum, the reaction was a little bit modified to use greener reagents. And there was some protein engineering done in Greifswald. And with the starting enzyme, or comparing with the starting enzyme, you already knocked down 88% of your carbon footprint. And as I said before, this reaction was really not mature enough to run an LCA. It makes no point. But we had the simplified metrics. We had the accelerated metrics. So we could actually pinpoint the pathway for improvement. We could chart the path. We know that the problem is in the downstream, although you can also improve the upstream. And if you recycle the solvent that you're using to extract the product many times over, that's how your footprint should improve. But not only that, once you've done that, if you increase the substrate loading of your reaction, that's how your metrics should improve until you reach something that is pretty good. And this can also be further decreased, and this is pretty good for a kilogram of product. Yeah, so key messages that I would like you to take is that finding and optimizing an enzyme is no longer expensive nor tedious if you use machine learning and microfluidics. With microfluidics, you can not only screen but also develop novel formulation that zeroes in where you want to release your ingredients. That enzymes, or I think we've demonstrated that enzymes have the capacity to synthesize many different ingredients for consumer products, starting from waste. So the toolbox is that big and it has that much potential. In our project, we do believe in the culture of metrics. No more blanket statements. Measure and then state. And finally, that smart and targeted communication strategy and dissemination strategy helps you reach better your target audiences and increase impact. Well, thank you so much for your attention. And I'll be happy to take questions after all the coordinators have presented.
Thank you, Aurelio. Next in line is the OXIPRO project. So now we welcome to the stage Gro Bjerka. She holds a PhD in molecular biology, and she is a research director at Norse Norwegian Research Center, running a research group in marine biotechnology. She focuses on exploration of marine resources and provide biotechnological solutions for the bio-based and marine sectors to promote sustainability and circularity. At the moment, she is the coordinator of the OXIPRO project. You may take the stage.
Great to be here. I'm so tall, I'm not sure where I should be to see everyone or for everyone to see me. At the core of OXIPRO, take some time, is the consumer products. I would say that's the core of OXIPRO. And we're using enzymes to get there. We have tried to focus on a group of enzymes that is underexploited, especially for this group of products. So we're harnessing oxidoreductases and their power to make changes in consumer products. We have been working on 4 different consumer product sectors: textiles, nutraceuticals, cosmetics, and also detergents. And you've heard many examples earlier. For the textiles, we have tried to develop a bio-bleaching process to lessen the impact of climate in this sector. And we all know that it's a very impacted sector. It impacts the environment. So we've tried to make a change there in processing of textiles, of the fabric. We've tried to use enzymes to use marine byproducts. I will tell a story about this later on to give you an example. And by that, reduce waste or unused byproducts, and also make sure that they are used for the right purpose. We've also used enzymes for producing new ingredients in cosmetics, especially sunscreens, and thereby reduce the impact of chemicals on ecosystems. And finally, we've also used enzymes to promote better hygienization so that we could maintain cold temperature laundry over time and thereby reduce the energy which is used in the process. So this has been the focus, the driver of OXIPRO. And we hope altogether to make an impact on very important goals of the world. The marine life especially is related to several. Water consumption is related to several, but for all of them is about responsible production. So this is the structure of OXIPRO, and you can see how the innovation cases are transversal for the entire project. We have in our matrix structure activities that are relating to responsible research and innovation. So we engage a lot with stakeholders. This is very important to get feedback during the work that we are doing. We have developed 2 platforms, which I will briefly present, one on computational work and one on, let's call it, wet lab work. We have also done scale-up of enzyme production to make sure that we get to the applications eventually in the so-called innovation cases that we have focused on. And we also have sustainability, so a sustainability assessment in the project. So I will speak quickly about 2 platforms. All the projects were expected to give something for the community, for all of us to work with. We have been working on establishing new data modules, such as ways to discover new enzymes. Machine learning has already been mentioned in a very pedagogic way by Aurelio. So we've used this to discover esterases, and this has been a very fruitful collaboration also with FuturEnzymes. And I think it's rare. I just want to voice Columb, and she was saying that it's been a strong collaboration between the project, and this normally happens on communication. This has been introduced by Nerea. It could happen on policy, but it also happens on technology, and I think that's quite unique. So we've also been engineering enzymes, adding new activities to enzymes, new functionalities, optimize the enzymes for the purpose that they're going to be used for. But this is one side of it. This has been ongoing for some time. We've made better and better programs, algorithms to do things. But now OXIPRO has also added these modules. Just a second. In workflows. So we're used a workflow manager called Horus to automate these analyses. So if you're not a computational expert, don't worry anymore, because these tools can make it easier for you. You can say you have a sequence, you want to see how it's structurally, how it looks structurally, you want to use AI. You could then paste in your sequence and you get out your structure from a workflow that is already established for you. So this automation of workflows, complex workflows, is now available through the platform, and this work has been led by Barcelona Supercomputing Center. We've also worked on various technologies for producing or discovering and characterizing enzymes, especially these oxidases. We've developed similar things to what Aurelio said, not as fast, not as small, but still useful, because these enzymes require cofactors. It's slightly more complex assays around them. So suitable for these discoveries, for these enzymes. So microfluidic devices. We've also worked on in vitro screening assays. And these have been used to identify completely new enzymes with new functionalities, like this peroxidase. And we've also described new versions of enzymes which has up to 30 different mutational changes to the enzyme. And this has also led, these new discoveries have also led to new patents, which, and in addition to a lot of scientific papers. So I will tell you about, these are the two platforms that should be of interest to a general public who are working on oxidoreductases, but also on other enzymes. And now I will try to present how the different work activities have been ensuring progress in a case. And I've been very fortunate to represent some of the other innovation cases earlier in public events, so I will focus on the marine byproducts and their utility today. So this is what we're going to talk about, the rise of fish byproducts. What are fish byproducts, you may ask? This is a salmon. It's a fish that I know very well from Norway, but I'll use it as a representative. So when we enjoy beautiful fillets and so on, we're eating about 50% of the fish, and the rest is remaining like trimmings, skins, head, viscera, blood, etc. 30 to 70% of the fish remains after it's been used. So what are we really talking about here? I had this yesterday. This is the Salmon Norwega on the menu. It's a pinchos that I had yesterday with a nice beer. Delicious fish, and I like eating it like this. It's a perfect fillet, it's a loin. But what is left, and which we don't think about, and this is the same for chicken, poultry, any poultry, beef. is the leftover products. But fortunately, there are very advanced modern biorefineries taking care of this. So these byproducts, coming here. These byproducts are processed in modern biorefineries by adding enzymes. We're not talking about oxidoreductases here first and foremost. It's like proteases that would cut the proteins. And by using that, you can do a refinement. You can see 3 fractions in this bottle. It's an oil fraction, very high value, and you see a transparent solution, and then you see the leftover, which is undegraded or insoluble. So that transparent solution is basically peptides and amino acids. They can be liquid or they can be dried, and currently they are used very much for our pets. So my dog loves it, I'm sure. But is it the right utility? These are food grade. Fish was food grade in the first place. These byproducts are still food grade. They have a very high protein level. And they all have all the amino acids intact. I mean, the industry has been dealing with these byproducts before, but they've used strong acids and bases. But this destroys the amino acids. So the enzyme technology preserves a better quality product. But the enzyme technology is also more costly, and therefore it requires a higher value market. The problem is, I think you've got it, they smell like fish, which can sometimes be a problem when you want to include it in a product. And these could be very useful, for example, as a clinical nutrition for elderly people who have problems to take up proteins. They could use these as supplements. So it is a higher value market, but why are we not there? No, because it smells like fish and it's transferring into the products. So we thought we could do something about it. We tested these protein powders, you can see here, from very many different groups, salmon, oyster, and sea urchins. And we find a lot of aromatics in these. It's not just one thing that smells, it's many things, many compounds. But one of the common denominators, which we also know from literature, is trimethylamine. It's a biogenic amine, which is naturally found in seafood. It's an osmolyte. So we thought we built a hypothesis in a prior project where can we then transfer, using an enzyme, convert trimethylamine that has this fish smell into an oxidized version, which doesn't smell. Like, this is the natural compound already found in very large levels in fish, but post-mortem, immediately when it's slaughtered, the TMA is turned into TMA. So can we revert this reaction? There is an enzyme type, we call it 3-methylamine monoxides, that can do this reaction. It's an FMO, flavin-containing monooxygenase, but as you can see, it requires a cofactor. I'm not going to go into that today, but we have been working specifically on the cofactor in OXIPRO. But this reaction, can it be done? Can it be done for this product to be able to reduce or remove the fish smell? That was one challenge we presented to OXIPRO. So I'm just going to show you one scientific result today, so I hope you bear with me. This is the, let's call it the concentration. And you can see here, if we test just the powder, so this is in fish, this fish powder, you can see a lot of gray here, independent of what temperature we run it at. And gray means trimethylamine, so that's the presence of this fish smell molecule. If we add the enzyme in a prior project, and I must say this is a collaboration with Manolo, we reduce the gray bars by about half. But you can see it works less at higher temperatures. So we need to have this process run to make sure it's hygiene and etc. So we need to run it at least 50 degrees. So we needed to make an enzyme that tackles that temperature. And so we did. So in OXIPRO, we made a temperature stable mutant. And as you can see, the gray bars are reduced, and you can see at high temperature, you get almost a full conversion of the fish-smelling molecule to something which is odorless. So this is an example of how OXIPRO has been working. But I think this is a reflection I think all projects can have. We can push technology, we can pull on technology, but what does it take for industry to adopt these new solutions that we are making? And I expected the other projects to show some CO2 reduction, greener processes, blah, blah, blah. I wanted to take in that other aspect of sustainability. Why is the industry not fully adopting these solutions? Why is not everyone using these wonderful solutions? So we had to look into some of the framework. We invited the social scientists in to look into what are the barriers for taking in this technology. And they find the access to the byproducts are extremely competitive. They're spread around the entire coast. It's extremely regulated what you can use it for. There are also high tariffs, meaning it's cheaper to send the full whole fish out, not leaving the byproduct next to the plant, etc. So we also looked into what can enable it, not just the technology. I mean, the technology is there, but what else is needed to sort of stimulate the industry to take advantage of this technology. So this is also a part of the sustainability that I think we should reflect on. These are examples from this particular case, but we have been very much involved with stakeholders. We've made material. We also engaged in round tables with the industry and with NGOs and other policy contributors to discuss the results and our technologies. We've been also very happy to see that the students are very interested. A fun fact is that these enzymes can also be used to make indigo or pigments, and we demonstrated the power of enzymes by having a denim staining course at a public school. We also have stands in fairs and expositions so that people get to know and understand the problem with the byproducts that are generated and what technologies can be used to reduce it. We've also had scientific papers and posters, and we also tried AI-generated podcasts to explain our technologies in very simple terms and to make it accessible for as many as possible. So I hope I showed you here that enzyme technologies is able to repurpose fish byproducts. We can make sure that they are taken from pet food to food. But it needs more than just the enzyme technology to be fully adopted. But if we do so, we can see opportunities for food production, for value creation, but also for sustainability on this sector. And that was just one story. There are 3 other cases. I could have told you a similar story, but I hope I have been able to show you how OXIPRO has been running, and that stakeholder engagement, computational work, the wet lab work, enzyme scale-up, fermentations at large scale, application studies, and then sustainability has all been driven by those innovation cases and the need for new, greener consumer products. I'd like to thank the entire consortium who has been all doing critical, essential aspects of the project. As the other projects have already demonstrated, it's a good mix of universities, RTOs, and industries, large and small. And I also would like to thank the other sister projects for a very fruitful, strong collaboration on communication, dissemination, policy, but also on technology. And with that, I invite you to take part in a survey if you want to contribute to the stakeholder work and have your say on the consumer products we have been working on, you may scan this QR if you'd like and join us, and you're more than free to contact us at any time. Thank you very much. I look forward to the question session.
So now we're going to introduce the last project for this first run-on sessions. So Manuel Ferrer is taking the stage. He holds a PhD in chemistry and is currently a research professor at the Institute of Catalysis and Petrochemistry at CSIC. His expertise lies in the discovery, design, and application of enzymes within the circular economy, and he is currently the coordinator of the FuturEnzyme project. Here we go.
Thanks, Nadia, for the introduction. Yes, sorry. So just before starting, so now I realize what the European Commission funded the 4 project. I really realized, because, not because we are good, but we are, I think, but because we cover many different things, different consumer products, different technologies, different enzymes. Indeed, I didn't see any single enzyme that is shared between the 2 projects. So it's a really complemented arsenal of looks, yeah. So it's a really complementary tools, enzymes, and product. And well, so now I'm going to discuss about the FuturEnzyme project. I'm not representing a single person, but a large consortium of 8 different academic groups, 2 clusters, and 6 industries, all with different and complementary, I think, expertise and interest. And I think the project has 4 different interconnected and in equilibrium key pillars, like the legs in a table that sustain everything. So the enzymes that we all know that has this superpower, they can make the reaction really fast and in the way we want. Then we have the customer that we all know that I'm pretty sure all of the people that are here today, so we are willing to pay for an ecological solution, but if they work. So I'm not going to buy a green product that is not working at home. So we prioritize also that the product is effective, and then the industry, because it's really committed to offer more sustainable and effective solution. And then we have the demand of the consumer. We are all the demand of the industry. And this is especially relevant for the product that we use at home. So textile, cosmetics, and detergent, for example, because their production and use is related to high water consumption, high energy consumption, and chemical consumption. So we need to lower this as much as possible. So with this objective, so we established 4 years ago the project, the FuturEnzyme, dedicated to develop enzyme-based innovation for making greener and more sustainable consumer products that were already in the market. And the idea was to discover, to put together a lot of scientists and industries to find a new and efficient and which is really relevant low-cost microbial enzyme, because we are working with consumer products that we are using at home, and then the cost cannot be high. So then the problem is, I think, was already discussed before. So it's a really challenge to find the correct enzyme and what we are dealing with. This is something that we need to pay attention. So, well, the idea is clear. So we can go for by prospecting campaign. We can go. This is a picture of one of the partners, Misha, with the comic-like style. Then we can take the material, we can isolate the DNA or we can isolate the microbes. So then with the help of computer, we can identify the enzymes. So then we go to the lab, we do a lot of experiments, find enzymes that do the job as we want, and then we produce. But so this is true. This is quite simple, but the reality when we target consumer product is not easy, because this is the case of finding an enzyme in laundry detergent. And then you imagine clothes, it's not like this because it's really clean, but you can imagine after playing football or something, so you have a lot of stain there. And imagine that you have a lot of fatty compounds. So you have long-chain triglyceride, you have long fatty acid ester, you have cholesterol ester that are in pigment or sebum. So then you have these parabens in the makeup. Then you have these phospholipids like in mayonnaise or eggs or something. So a single enzyme should be able to degrade all of these things, just to be removed during the wash. And the most important, and we learned from Henkel, that I didn't know. So it's nice that if the enzyme do not work with a small ester, because they smell. So if you add an enzyme that do the job also cutting this small one, so when you open the wash machine, so then you will not going to the party with friends, because so it's really a challenge to follow this. And then the problem is we cannot work with all the detergent. We cannot speak an enzyme for the detergent because each detergent has the different composition. We can think that the enzyme should resist in this condition. Then it should be stable under different storage conditions, because it's not the same having the detergent formulation in Madrid in the summertime, 45 degrees, or in Norway with minus, I don't know, 10, or I don't know, in the mountain, you take to the mountain, and then so it should be resistant under different storage conditions, and then under different washing conditions. So that means temperature is, we prefer, and this is what the partner are asking, so enzymes that are active at 30 degrees, so we reduce the amount of electricity in the wash machine, but at least some people will use 60 degrees, so 50 degrees. So the enzymes should be acting in different conditions. The water that we are using at home may have different pH, and then we have the time, because sometimes you put the wash machine 15 minutes, but then other 1 hour. So the enzyme should be stable on this. Well, what happened with the textile? And this was Schuler, our industry partner, asked us. So while producing the fabrics to finally make what we dress, they use what they call spinning oils, and these are the chemicals that are used. They are in these roles during the different transformations. So we need to remove all of this. Sorry. Which is not an easy task, because we need to find an enzyme that do the job. And what is more important, and also where we will see the video. So this is the fuller machine. So where they transport the fabric in just to use hot water, a huge amount of hot water, just to remove these chemicals. And this is the fuller machine, and they are asking for an enzyme that do the job in 30 seconds. So this is on the right side. You will see how the fabric is coming. So the enzyme is in the middle. The enzyme solution is in the middle. And then each sample goes through the fuller machine twice. You will see it's just 30 seconds. So it's incredible. So it's really a challenge. It's a challenge to remove these chemicals, and also it's a challenge that just in very short time. So it's the enzyme is doing the job. And then we have the cosmetic, which is another target product in request by Evonik. So we have high molecular weight. So they produce high molecular weight, and then using a well-established industrial process that they have. So they are able to produce this hyaluronic acid HA50. So it's used. So it's a real product that finally is integrated into a product already in the market. And then they request just to cut this high molecular weight hyaluronic acid using enzyme at room temperature and in water. So no solvent, no thermal conditions, anything, just to cut in a specific size, a small one, not really big, not too small, because the properties will not be good. So in a perfect size that they are asking for, and this is not easy. First, because there are not so many enzymes that do the job. Also, you will see now later on that it's a really viscous solution, and then it's difficult to do this transformation. So with all of this in hand, so we realized that we cannot speak about finding an enzyme for detergent, because each detergent has different composition. Or we cannot speak about the hyaluronic acid, because you can find in the catalog many different types of hyaluronic acid with different sizes. And then we cannot ask about textile in general, because there are many processes while we have the raw material to the final thing. So at the end, what the partner Henkel, for example, gave us the formulation that we need to make greener. Ebony shared with us the hyaluronic acid, high molecular weight, hyaluronic acid, and the one that to benchmark that they produce and they have on the market. And then we use the fabrics with polyester or polyester with elastane and so on, that with the spinning oils integrated. So we can use real product in the market. And then we need to find enzyme. So our approach was really clear. Well, let's do a massive bioprospecting. Let's go to all over the world, check the current public databases, and take all the genomes and metagenomes from our projects and public ones. So we cover about 800 different site explores. So we take all these genome and metagenome sequences, we isolate new microbes, we sequence the microbe, we take the genome, metagenomes, and so on. And with all this information as a backup, so then we made the screening of the enzyme that we are interested in, mostly lipases and hyaluronidases. So first, which is really what was really crucial. So we got the feedback from the industry partner. They told us the composition, where the enzyme should be stable, where the enzyme should be working, the chemical composition of the detergent, the molecular weight of the hyaluronic acid, and the composition of the fabrics. Well, with this in mind, so we use bioinformatics tools to identify at large the enzyme, and also we apply computational filters. And this is our partner made a great job. So do computational analysis to predict which of the enzyme can bind and convert the chemicals that we would like to convert or degrade or hydrolase, and also check the stability determinant of this enzyme. So we are looking at the enzyme that we would like to have at the end. And also we have engineering platform also aligned with product needs. For example, if we need a hyaluronic acid, a mini hyaluronic acid without enzyme in the final formulation, why not to use, for example, supramolecular engineering just to immobilize the enzyme, so we are sure that, for example, there is no single molecule of enzyme in the final product. This is just an example. And then, which is crucial, is just we need relatively cheap enzyme that can be produced in the quantities that industrial partners are requesting. So then our industry partner, Biosense, is having this pipeline for producing in pigia, because we need extracellular enzyme. So we normally, all the enzyme were produced at 10 liter scale as a first... thank you. And then we normally produce at 10 liter scale, but then later on, to make the LCA assessment and to have more material for the future, so they were producing at 1,000 liter scale with jar scale. I think RadicalZ already mentioned that they have kilogram scale biocatalyst, so we are also interested in producing this. And this is just one example. We do in collaboration with the pipeline. So Professor Fernandez Lobato and Dr. Marina and Fadia, which is here from the lab in the audience. So basically, we do the hyaluronic acid. We solubilize in water. Then we add some amount of enzymes. And then we just, after a while, and at almost room temperature, so we separate the enzyme, and then we lyophilize, and then we got this material, this mini hyaluronic acid. And this, just to, I'm not sure whether the video, you see here on the left the hyaluronic acid, it's really viscous, even the magnetic still cannot move. But then after the while with enzyme, so you see how the hyaluronic acid is hydrolyzed, and then by HPLC, so we can analyze when the enzyme cut the product to the site that we want. So this is what, and I didn't, I cannot go back. Sorry. So the interesting thing is that we were able to at least minimize by 30% the emission during the production, okay, of this mini hyaluronic acid, because it's something that you can do at room temperature and with water, so nothing else. So which are the legacy of our project? So these are, as Aurelio mentioned, numbers. So we bioprospect a huge amount of environmental science. We were able to retrieve for future generation, not only for us, but future generation, new enzymes, sequences, products that were produced by biosynth of enzyme material that can be commercialized, for example. And also we have some digital material, because our computational partners do some, in collaboration with OXIPRO, for example, they make some web server for enzyme screening. And also we develop a number of automatic engineering tools that can be indeed be used by future generation. Well, which also is our legacy in the findings, and I think this we discussed also in the frame of the cluster. So by consumer survey, we know that consumers prefer greener product, but if they work well, this is important, and this is was something that we learned from our surveys, that enzymes are safe. And this is important because we just published this policy brief within the cluster, where we try to convince the regulation bodies that the enzymes are safe. And indeed, our industry partners are aware about this regulation, and they try to tell us that it's 100% safe. So we saw that enzyme technology in some of our case can cut emission by 30%, but, and this is important, enzymes still contribute to environmental impact. It's not that, it's not something like we are happy, but the enzyme has zero effect in the environmental impact of the final product. So we need to, as little enzyme we use in the final product, as better. So we need really effective and stable enzyme. So when you do the LCA analysis for your enzyme production, you consider everything so that enzyme is produced with the minimal environmental impact and with the higher performance as possible. So you use as less enzyme material to the final product, so to minimize this environmental impact that in some cases, like in the detergent, is really big. Also, we learned about this enzyme innovation. We reached, without problem, I think, most of us, TRL 4.5. And recently I was at the CSIC in a conference, and there was an expert in TRL at the CSIC. And they were telling TRL 1, 2, 3, 4, 5, 6, because this, it really takes time. It's everything not really fast. So this 4, 5, 6 is really, really long. And then we realized, for example, with the Bonnet, that it takes some time to make all of this toxicological analysis, thinking in cosmetics, formulation stability, and so on. So we need extra time. The discovery must match industrial needs. So if we are developing consumer product, we need to work with the consumer product that we have to implement, to make green and sustainable, because others detergent may have different compositional requirements. So this is also important from our project. Only a tiny fraction of the enzyme that we discover, only a tiny fraction, are used by our industrial partner. So the rest of the enzyme we cannot keep in our lab. So somehow we need to offer to others. And this is, so we are trying to make license agreement in order they can be, you can buy on the market. So this is because probably other companies will be interested in this enzyme, because we have seen that this enzyme has a cross-sector potential. So the recommendation, and this is following also a policy brief that we published recently. Yeah, I need to fight with the, did I have the here? Yeah. So we need to recognize enzyme as distinct category for normal chemicals. So enzymes are chemical, but it should not be put together. We need to clearly show the benefit of the enzyme, and not only by LCA, but also the benefit of the innovation product that we produce. We need to ask the funding agency to provide funding. and especially a way to better implement the enzyme technology as fast as possible, with less regulation, as Gro mentioned, as possible to go to the market. Always being sure that the technology is safe. We need to include biodegradability and low toxicity in enzyme evaluation criteria, promote sustainability enzyme innovation through financial support and collaboration. This is clear, and industry play a higher role. And from my opinion, we need to convince national or European people like the money that we need to fund pilot project and demo plans for enzyme-based product. For example, if at the end of the project we reach TRL 4, 5, 6, somehow they need to open window to fund this project to go further. If not, the enzyme technology will be stopped at the end of the project. And this is, I think, we consider also important, create EU-wide hubs, I don't know how to say in English, for enzyme innovation and extractive technology. So at the European level, we need to say, well, the enzyme technologies are used for all these products. All these products has different characteristics. So, well, let's see which enzyme we are thinking of, which are the characteristics that this enzyme should have, and do something together. And also thinking in the independent of Europe. So Europe must be independent of now in the global situation. We need to be independent of enzyme technology from, for example, USA or other countries. So we need to create these hubs or connection network within Europe, so we are clear that we can develop enzyme technology independent of other probably third parties. So just to see where we are. And this is the video. It's just 1 minute. Our project. This is just to. Yeah, so that's all. And just the final word. So thanks for the. I mean, I agree with Gro. So the work with the cluster was really successful. For us, was a really successful work we have done together. And then that's all. Yeah, for question now. So. I mean.
So maybe our previous speakers, you may come to the stage as well. Yeah.
Yeah, I know.
Sorry. Mine stays in place.
Okay. Any questions?
If not,
You have one.
Very silent.
I do have a question for all of you. So, as I said, on behalf of the Commission, I have been impressed by the work that each of you did and the work that you did in the cluster. But you know that at the Commission, we have always this question: what happens at the end of the project? And do you have any ideas of how to make this cluster live after the end of all of your respective projects? Do you have any plans for that? I mean, that would be very nice to hear. Thank you.
We already discussed this a little bit. And the interesting thing, I mean, I had a collaboration with him before OXIPRO, so we go back a long time. And I've learned to know Aurelia during this project. We've established new projects. So I think there are collaborations between the different 4 projects living in new projects. I mean, this is one way of taking collaborations forward.
Yeah, I think in my case, like I mentioned at the end, so although some of the projects are finished, so I think this should be the starting point of adding more and more, so we can create a bigger cluster, just to convince. I mean, if you give some different views to the same solution, it's an enzyme solution for enzyme technology for something. So if we enter to the cluster different views, different activities, different validation data or experimental data that support your claim, I think will be good. So I think this is a starting point for the future.
Maybe it's the opportunity today to speak about it with all partners, to meet them and to also plan the future. I think it's like a nice opportunity to have this meeting today.
And speaking from or on behalf of ScienceSeed, it's true. I mean, it was chaotic last week with lots of emails back and forth, but in between those emails, there were also proposals to keep the newsletter going, and from our continuing projects, so to join and build critical mass in tasks of communication and dissemination.
Yeah, if I should add to that, we are also, of course, aiming for new products and new projects and new collaborations. But along with that, we have at our university taken a strategic decision to also go for a little bit higher scale. So to adjust a little bit the research profile in order to be able to move the research to the pre-piloting part.
Can I add that I love your idea of an enzyme hub as well? It would be nice to invite more people than just the 4 projects or associated projects. It's very vulnerable if we don't get the next grant or whatever. You fragment your network. So if we could have Commission or the community engage and make an enzyme hub or an enzyme cluster, this would have been more long-lasting, more valuable for everyone, I think.
Yeah. I think that's a wonderful, actually a wonderful idea, because we also see from many industrial partners that there is sort of a return towards interest in enzymatic processing that is regaining sort of more and more momentum. And I think that has to do with, I mean, we've seen a lot of examples of that here and now, that we are having more and more information technology that can help us in our discovery routes, and to combine this with the sort of processing technologies. I think that is a very valuable route to go, and have this sort of hub that you suggested. I think that would be fantastic.
Yeah. Question? There? Somebody has a microphone there? Or, yeah, back of the room.
Thank you. Hi, thank you. I just wanted to ask you about the role of AI in all of your projects, and how do you see the future of the use of AI and machine learning in the bioprospection of enzymes, and how this is going to improve more, and how do you see the biopropagation of enzymes in the future, like in 10 years or 20 years?
Yeah, thanks. So
Yeah, sure. I'll begin. I think what we're seeing is the beginning of AI for bioprospecting and enzyme optimization. I think you've seen successful cases and two different approaches, maybe from the public sector, and in RadicalZ, we approached it from the business or the private sector point of view. You see that it's functional, and I think now is when we are going to start finding the true limitations of it in the coming years. And it'll be a roadblock solution type of development, in my opinion. Now, coming back from the opposite side, from the functional discovery side, I still think that there are properties and activities that are harder to predict by machine learning. So I think there's still room for just about everything. But don't get me wrong, it should not be an either-or decision, but rather an and decision, right? There are tools in the toolbox. Use all of them.
Yeah, I think that, just as you say, AI is coming. It's developing everywhere, in all types of sectors, and both in design and processing and evaluation. And we are in a phase where there are not too many tools available yet for those who are not developing the AI. But I think that's where we are ending up in after some years. We will have tools that allows the use even for those who are sort of not the primary developers of the different programs or tools. Being somebody who works with enzyme, you just see how much it has influenced structure determination after you having this AlphaFold tool that was developed. So I mean, you can see how it's more and more sort of developing into different areas. Yeah. And I think that's something that we should embrace and use.
Yeah, I can. So thanks, Rafa, for the question. Yeah, I think the AI is already the present, and for sure the future will be much better. We are not synthesizing protein if we don't ask first to the computational people, because it's really time-consuming synthesizing. So we need to ask. So, but they need also metadata. So the future of AI of machine learning is really linked to the metadata, experimental data from the project that we are running now and future projects, so they can do better prediction, better and better and better. So the future of AI, from my point of view, is this. So the tools are there for sure. We were discussing yesterday with someone. So the tool that we used 6 months ago has nothing to do with with the tools AI tool that we are using today. So they are changing so much that for sure the future is really will be the key. But also always dependent on experimental data. But this is my as a experimentalist. So.
Fully agree.
But just about AI, I think we should very take care of the data that we supply to AI. It is something very important, because when you share data to the AI, you share it to everyone as well, and the AI will learn also from that, and we have to be aware of that, which could be maybe problematic. It could be problematic for IP protection and so on. So this is really something that we will take care of. So.
Yeah, I'll just make a note that the tools that you refer to, they came during the life of these projects. So it's fairly new, and sort of during these projects we've learned to use like AlphaFold, which was mentioned. There are many other tools. And we thought we had solved the last crystal in the project. It now feels unnecessary to do it, but for at least for our purpose. But it obviously isn't, because crystallization may still have a role. But we use these tools to the extent that they have been developed and accessible. But we see it's coming on all areas, on genetic design, but also I would say on the applications on process control, process understanding, especially involving very many, a complex substrate, a complex product. We see AI also being useful for us to understand those complex processes and sort of how to stop a process, how to control it.
Yeah. So, and then just a final remark. When we, a few years ago, we published a review, and then while reviewing the papers, I realized that there were some people quantifying the amount of CO2 that we produce in the discovery process of the enzyme. So they were able to quantify the amount of CO2 that you invest for doing the experiment, the amount of CO2 that you use while using the computer. Soon they will quantify the amount of CO2 that you need to take if you are working with the computer to go to take the coffee and come back and so on. So they quantify that the computers and the machine learning has a cost for CO2 production. So we need to optimize the IA machine learning tools so they can do this screening process, not only in the optimal and 100% validity method, but also in the shorter time as possible, so to allow the lowest production of CO2 during this process, because computers also account for CO2. So as much better tools, as better, yeah. Mm-hmm. Perfect.
Mm-hmm. This will be the last question because we have scheduled the coffee break. Okay, so, but please go ahead.
Well, okay, then my question is going to be shorter because the coffee is important, I think. No, the point is about your point of view about regulation. In terms of in Europe, we love regulation. We are a lot of countries, national regulation, and everything that contains DNA, of course, it may be, or it is a public concern. For example, I don't know if talking about enzymatic reactions could be associated with allergies for the public media or something like that. What is your point of view about where we are going? And if you just develop an enzyme that works here, maybe in the next country that it will work also, how much it will take to have a common policy about that?
Yeah.
We worked a lot on this. Yeah, it's a good question. I mean, being an enzyme, being a protein, can pose allergic reactions to a worker who is exposed long-term, uncontrolled. But as far as we know, I mean, we're your industry, but the industry has procedures for this to control the exposure. And we have been discussing, there is this potential risk using enzymes. I think that has to be transparent. This is regulated. It's called a respiratory sensitizer. But there is all these sustainability benefits. So we can't have that safety issue being a cutoff that would not allow enzymes in use. And what is the cost? The cost is the sustainability benefits that doesn't reach the market. Mm-hmm. So I think navigating that it's complex, but one way we could do it is to provide guidance on how to work with enzymes. Mm-hmm. As that's one alternative. Instead of banning and regulating, we could rather say, okay, there's an exception for enzymes, there's special chemicals, biochemicals. This is how you could work with those specifically. So they are essentials, I would say, mm-hmm. and thereby requiring maybe, well, not an exception, but a different treatment.
Mm. Yeah, we've been discussing a lot about the regulations also, and one important aspect that has come up in many of the discussion is the time. If there could be a shorter time from your sort of put-in of an application to have a new process or a new enzyme to the sort of answer from the regulatory bodies, this would be highly beneficial. So there are many people that are applying for using sort of novel type of in processing tools, etc. And a big limitation is if the timeline is very long until you get an answer on your application. This is problematic also for the commercialization aspect.
Well, perfect.
Anything more?
Yeah.
Okay. So the sessions will continue at 12:45. Until then, you may join us at the coffee break. Thank you for all the speakers so far.
Thank you.
Thank you.
Hola. Hola Mafalda, ¿qué tal? ¿Nos oyes?
Yes, I can hear you.
Okay, perfect. We are the technician.
Okay.
Okay.
Speak in Spanish, I understand. I don't speak it.
Okay. And you have we have a presentation here.
Yes, but I also have it on my screen.
Oh.
If you prefer that I share it.
Okay, if you want to check to share now.
Try to share it. Do you want me to share it?
Okay.
Okay.
Well, perfect.
Yeah.
Yeah.
Let me see if I can change the. Okay.
Yeah, perfect. We can see.
Let me go up. Okay.
Well, if you want to share now or yeah, it's here with the cam, and when you want to share, you only
Okay.
make the
Just
same.
to be aware, when on screen, will I be my face will show to the participants or not while I'm speaking?
When you are speaking, we can see the slide, and we are the cam in a part of the screen.
Okay.
We can see the both.
Okay.
Okay, perfect. Thank you.
Do I leave it like this now?
Well, if you want to put off the cam, but not.
Yeah.
Okay. Well, perfect. Thank you.
Okay, thank you.
So, welcome all back to the sessions. As you may know, now we have 3 different roundtables on different aspects around enzymology and sustainable consumer products. But first, we have an intervention. Joining us online will be Mafalda Quintas. Mafalda Quintas is a policy officer at the European Commission's DG RTD Healthy Planet Directorate, focusing on green transitions and bio-based systems. She's involved in shaping policies around biotechnology, biomanufacturing, the development of sustainable bio-based industry. She supports the Circular Bio-based Europe joint undertaking as well. With a extensive background in science policy, management, and innovation, Mafalda has worked in numerous interdisciplinary research networks, fostering collaboration between academia, industry, and policymakers. Before joining the European Commission, Mafalda worked for the COST European Cooperation in Science Technology, where she led the development of evaluation procedures and grant management systems, enhancing research collaborations across Europe. Her research expertise spans from food science, bioengineering, and sustainable technologies, with multiple contributions to bio projects and scientific publications. Mafalda has a PhD in biotechnology from the Catholic University of Portugal. So, welcome, Mafalda.
I don't know if you can hear me?
Yeah, we can hear you.
Okay, thank you very much. Well, good morning, good afternoon to all of you. Let me start by thanking for inviting me for this short intervention, and also to apologize for not being present and following your entire event, but I've been following some of the discussions online this morning, and I want to start by congratulating the community for all of the work that has been done in these projects. But today my focus is a little bit on giving you and providing you a little bit of perspective on policies that are supporting a sustainable future or the green transition of our economy. Let me check if I can change my slide. Okay. It was working. Okay, yes. So starting a little bit from the top, last summer, President von der Leyen set the priorities for her second mandate for the period of 2024-2029. And we have a new focus on competitiveness, but it's a renewed focus on sustainable competitiveness. The idea and the objective is to bring a new era for European defense and security, but also support the citizens, our social model, our quality of life, and our values. Of course, at the same time, keeping the European leadership and influence, and also openness to global cooperation and towards the simplification of the European Commission processes in the different aspects and files that the Commission manages. By the end of the year, we had the new college starting their mandates, and we have a new commissioner that is Commissioner Zarajeva. She's focusing on, she has a different title. It's the first time that the commissioner in charge of research and innovation also has startups in her title, and this is a significant word, because she has several I'm sorry. I think there's been I think there is a problem.
Hi, can you hear us?
Hello? I don't know what happened here. Hello?
Hi.
Okay. Okay, so something, can you hear me now?
We could hear you the whole time, I think.
Okay, sorry, because there was some something that started playing on the back of my computer. I'm really, really sorry. So I don't know where did I stop my intervention, but then, as I was telling, the new commissioner is commissioner not only for research and innovation, it's also for startups, and this has an important aspect of her priorities. And the first priority that are of the new commissioner is to strengthen research ecosystems. Of course, this is a point that will be coming up in the ERA Act that will be put forward later. The second big priority is to help unlock the European Union innovation. There are still a lot of obstacles facing innovators. I heard a little bit some of your discussions in the morning, and so you are also aware of some examples of what are the things that actually are not allowing to a smooth transition from the lab to the market of the best European ideas. For that, there's going to be an European Innovation Act later in the mandate, but we will have an EU startup and scale-up strategy, and this is a communication that will be coming out in a couple of months. The third is to boost the European capacity in key technologies. You have been discussing the importance of AI for developing the science. It's not going to be substituting experimental, and is not going to be substituting the human brain, but it's going to be a very important tool, specifically also in your area of research. So there is an increased focus on looking on how AI can actually help support and boost the efficiency and the use of AI in science. So the fourth priority is about to build an inclusive research and innovation union. And next year, the Commission will present an action plan for women in research and innovation. And finally, delivering an ambitious successor for Horizon Europe. We know that we have a very, very strong research and innovation funding program in Europe for many, many decades. And the next one will build on the successes and the lessons learned from the past. Going a little bit into the, going a little bit on the policy landscape, we are facing, I think that this is becoming over a little bit of a commonplace, but we are facing an imperative and pivotal decade. Things are changing every day, and we need a systemic transformation in our society and in Europe. But President von der Leyen, she was very clear in her mandate, is that we stay on course of the European Green Deal. But of course that in a period of global realignment, we have a new focus on competitiveness. We have published the Competitiveness Compass, and this compass gives us a very, very clear destination where we aim that when we say that Europe must be the place where tomorrow's technologies, services, and clean products are invented, manufactured, and marketed. And this competitiveness compass does not only give us this destination, but also gives us a clear roadmap of how we can achieve that. You have seen probably the recent communications on the Clean Industrial Deal that aims to promote sustainable industrial practices and reduce carbon emissions, while of course fostering on economic growth. But we also have put forward a bold vision for Europe for agriculture and food, aiming at boosting the agri-food sector, but attractiveness and competitiveness, but also providing a clear statement on the importance of the agri-food sector for the bio-based industries and for the bioeconomy. So during the 2025, there's going to be two important communications coming up. Is the life science strategy and the bioeconomy strategy. And later, at the beginning of the next year, there's going to be also another important legislative initiatives that will actually have, that are also related with the work that you do, that is the Biotech and the Circular Economy Acts. Looking a little bit to the life science strategy, the life science strategy is going to be presented this June, so already this year. It will aim to support the development of uptake of safe technologies that boost competitiveness and prosperity, accelerate the green transition and digital transformation. So the focus is on innovation, growth, and impact across Europe. The call for evidence is open until the April 17, and I would really encourage you to submit your views. You have prepared already very good policy recommendations, so you already have a lot of information that you have collected based on your work, on your science, on your scientific knowledge. So I really encourage you to translate these recommendations that you already achieved in your projects into providing evidence to the life science strategy. Also a little bit focus on the bioeconomy strategy. The bioeconomy strategy was first released, for the ones that maybe know, it was first released in 2012. It has been updated in 2018. It has become kind of a fil rouge for the development of the European Green Deal, and it helped in the green and just transition of the European economy. And we will have a revised bioeconomy strategy that aims to promote more circular, sustainable production, use and consumption of biological resources in a sustainable way, and for food, materials, energy, and services. So it's something that will come at the end of the year. There are the bioeconomy strategy. There's the plan to be built around four main challenges. The challenges on how to scale up, something that you have been also discussing a little bit on, that is somehow currently, that you somehow are able to achieve TRLs 4 and 5 in your projects, but now how to bring it further and closer to the market. It will be also looking at how to source biomass and how to find alternative sources of raw materials within and respecting the planetary boundaries. Creating also some market demand for green and clean products, and also looking at international dimension of the development of the bioeconomy and also the green diplomacy. There is also a call for evidence ongoing. This one you have a little bit more time. It was open on the 31st of March, and it will be running till for 12 weeks, meaning that will close on the 23rd of June. Also before continuing, I also invite you to keep a look on the call for evidence that will open for other initiatives, for example, like the Biotech Act that will be later. And I think that you really have a lot of science-based knowledge and science-based opinions, and this is something that is very much appreciated. So please keep an attention to when will this call for evidences open, and please provide your input. And a little bit on research and innovation. Of course, that we cannot achieve a green transition only with policies and policy initiatives. We really need you, we need your ideas, we need research and innovation to develop and bring to the market the bio-based and the bio-solutions that we are looking into. I invite you to learn a little bit about the funding opportunities that are upcoming. So the Horizon Europe Cluster 6 Info Days will be organized on the 20th and 21st of May online, so somehow easy to follow. In this Info Day, there's going to be the presentation of the topics of the annual work program of 2025. So so something that can be of great interest for you. And also just to remind you that the CBE, the Circular Bio-Based Joint Undertaking, has currently the call open. The deadline for the call is 18th of September. The current budget is 172 million euros. And what I would like also to mention is, also following up from your discussions, that there is a big focus on biotechnology and biomanufacturing in the CBJU calls, and also they have funding for higher TRL levels. So in CBJU, some of the topics are for innovation actions going to 7 TRL, and some flagship initiatives that go up into TRL 8. So I think it's important for you to be aware of this. Finally, also a little bit, it's coming a little bit from your discussions on how to sustain your network, on how to keep the cluster alive, and how to spread the cluster and become maybe a network. Just because I worked for many years for cost, just look, there is an open call every year in the cost program that can actually fund networking activities, and it's a good instrument to structure research and innovation communities. So in case you are interested in growing your network. Just before, I'm just about to finish, I think I am a little bit over my time already. Let me emphasize a little bit as a final message the importance of the alignment of research and innovation within the broader EU policy framework. And I would like also to invite you to think about yourselves as the drivers of a journey that we have as a society, as Europeans, that we have to take. This green transition journey, it's one where each of your ideas, each of the projects that you manage, each of your innovation are contributing to a sustainable, prosperous Europe. So thank you for the work that you do every day, and please continue doing it, because research and innovation and scientists and innovators are what brings us and makes us a better society. And I thank you for listening to me, and I look forward to continue hearing from your community in the future. And if you have any questions, please don't hesitate.
Thank you, Mafalda. Does anyone want to have a quick question to Mafalda before we start the roundtables? Maybe I can let you this mic.
Mafalda, so many thanks for the presentation and for sharing with us these ideas and information about the new action plan. So I have two short questions. So first, thank you for suggesting this cost action to continue the cluster. We didn't think about this, so thanks for the suggestion. And my main question is the following. So if I see, we are thinking or we are discussing today enzyme technologies, but if I see the Cluster 6 program, enzyme technologies are somehow not really a priority there. So do you think the next action plan, this enzyme technology, will again be a priority compared to the Cluster 6? Or, yeah.
Well, I think that the life science strategy is going to be a strategy that has a lot of research and innovation. Of course, it's not only about research and innovation, but has a lot of research and innovation embedded on it, because it's really about how to bring life science technologies to the market, to use them for new products, for new processes that are cleaner and that make better and more innovative products. So I'm convinced that we will see, because of this strategy, like we have seen for the bioeconomy strategy, that for many years was somehow providing some frame to the development of the bio-based sector, we will see in the life science strategy likely the place where the life science technologies, and this of course includes enzymes and includes, for example, fermentation and all these types of technologies. I think that we will see more attention to these topics.
Thank you. Okay, so let's move to the first of our roundtables, which will be on sustainability and research projects. Thank you very much, Mafalda, for your intervention and for joining us in this event.
Okay, thank you very much. Bye-bye.
And introducing our next moderator for the sustainability in research projects, we have Mona Arnold. Mona Arnold, the moderator of this table, has more than 20 years of professional experience in innovation and R&D management in national and international projects relating to environmental technologies and environmental affairs across various industries. She's especially acknowledged in sustainable businesses, resource efficiency, circular economy processes, EU waste, and water policy tools and the twin transition. At BTT Technical Research Center of Finland, she is currently working as principal scientist in the team's sustainable business models, with the responsibility on solutions for circular economy and digital transformation. Thank you, Mona.
Thank you. And can I ask all the participants to the panel, please come here and help me out. Okay. So I have good morning to you all, or good afternoon already. I have the really the honor and pleasure to facilitate this next 30 minutes. So that we're going to stay on sustainability. And I have a little bit of a double role here. I'm also representing the OXIPRO project, where I was managing the work on sustainability. So a couple of words about that. Then could you please put up the first slide? On sustainability in OXIPRO. So the aim there was to provide feedback, with the work to provide feedback to both the enzyme developers and also the product designers. We were looking at the whole scale from the raw materials to the end product. And so we had a bottom-up holistic approach with a very quite holistic multi-criteria point of view where we evaluated the 4 innovation cases. We were working on detergents, textile biobleaching, nutraceuticals, and the sorry, I have a blackout here now. Sorry. Yeah. And so we looked at it from the 4 pillars of sustainability, the environmental, social, and economic sustainability. We also were assessing the circularity of local value chains and providing there or developing roadmaps for circular economy around 2 of the innovation cases. And then also looking at the risks from the toxicity point of view, and that was done on the detergents. It was very interesting work in the sense that when we looked at the product, the end product and its whole value chain from the cradle to the cradle to cradle, I would say, or cradle to gate, then what we actually found here, what was important, I think, for the scale-up is that we were able to look at find the hotspots. Where can you have the biggest impact on sustainability when you look at the new product? And then in the end, what we saw also that it was actually not the product per se necessarily, but it was what the enzymatic, the inclusion of enzymes actually could enable that made the whole process, the whole application then more sustainable. That was like one thing that we found. Or then also that actually, yes, the product is more sustainable, but it requires some modifications to the packaging, and then we have the load from the packaging again that sort of like balance out the effect of or the beneficial effect of the enzyme. So I just call for it's really important to look for the whole, not only the product but also the application and what you actually can receive from the use of enzymes. So that's all from me, and then I go over to, no, I keep quiet, I hope, and I go over to my peers here. So please, we're going to start with Monique and without slides, and my question is like, how could you describe with a couple of words how you've taken on sustainability in your project? And start with your own presentation first. Who you are, Monique van Lochem from Evonik.
Yeah.
And then how do you see that it has helped scaling up the research and innovation results in your own project?
Mm-hmm. Yeah, thank you.
And then it's Pablo, and then it's Heiko.
Yeah, okay. Yeah, thank you for the nice question. We as Evonik, we participate in the Future of Enzyme project as an industrial partner in the consumer products, so cosmetic applications. And we were very much interested in an enzyme that could produce our active cosmetic ingredient Hayeke in a more sustainable way. And even if we do not use the enzyme in the final product, it is very crucial that in the production process is as sustainable as it could be. And the use of enzymes is here extremely important because the current process is very conventional. We use a lot of steam and electricity and basically electricity and steam. And if you can use enzymes, then normally you can have a process much more, let's say, gentle temperatures, and environmental impact is so much lower. So if we can produce our Hayeke with enzymes and it is more sustainable, that will boost, let's say, our product, and our consumers are also even more and more interested in more sustainable products. So it is really important. And we were very happy with the results in this project and looks very promising. And at the moment we are in a, let's say, pilot scale and testing the product, but we are very convinced and very happy with the results so far.
Thank you. And Pablo, yes.
You have the slides there, but do you have the pointer or something?
Yes, it's up here.
I will say next one and then you can go, okay. Good afternoon then, yeah, please click.
You wanna?
Okay, so I'm in.
You wanna come here then?
Yeah, I'm going to come here because otherwise I don't see. Okay. Thank you. So I have been involved in RadicalZ on the environmental part of the project, and then the first thing that we realized was that we had quite a lot of different approaches and projects inside. So the first question that we had to address was, depending on the process that we wanted to have, we needed to define what we were going to measure. We had some industrial partners that made quite clear from the very beginning, we want to start from here, so we know already where the things are going to be made, which is the raw material we are going to use, where this is going to be, how the process is going to be. So for this type of approach, we could enter the full LCA. So we tackle all the things. But then we realized that there were quite a lot of other processes that were at TRL 2, 3, starting new molecules, new enzymes, new reactivities with a lot of interest too. But from these processes, we couldn't make, it was not kind of meaningful to try to make a full LCA because we lack quite a lot of data. And then we thought it would be great if, it would be great if we could make a kind of a method to make this study here more in a narrow way. And since we are in RadicalZ, as Aurelio said, we have to be rapid. So it has to be a rapid method to measure things in a rapid way. And then we thought, okay, why don't develop a method to measure the CO2 that is being formed in enzymatic reactions, say at the very beginning, gate to gate, but only using the things that you have on your reaction. So you make a reaction, you know the loading you're going to put in substrate, the reaction time, the temperature, the conversion, and so on. So no other kind of values that are there, but we don't know where they are, you need to look for, but just things that you have on your own reaction. And then we got some kind of system from the bottom up to make these kinds of things, to try to measure in a rapid way how the global warming potential of the reactions is being take place, because this is giving us the chance to know where we are and how to improve the things. So quite rapidly, first we need to measure which is the CO2 impact of the treatment of the solvent and water that we are using. And this is followed by this equation, which means this F means depending on where you are using a solvent or you use a water waste, you need to put this factor. So if you incinerate the water, you will have more impact than if you simply make the water in the wastewater treatment. If you incinerate an organic solvent, of course, it will create more impact. So depending on the F that you have here, there, you will have this value. Conversion and unloading is a denominator. That means that process intensification is key. The more loading you put, the higher conversion you get, the less environmental impact you are going to have. This is one of the lessons that we got in RadicalZ. It's quite important to have this. This is only half part. You have a second part because we need to think in the energy that you have to put in the reactor. You have to heat the reactor to make a process, usually even in biocatalysis at mild temperatures, but still you need to heat. For this, there were also two equations. One is for the energy if you use an organic solvent, and the other one for the energy if you use water. The energy you use in the water is higher because of the higher heat capacity of water, meaning that you are going to make more CO2 out of this process. You can measure with these parameters. And also, I don't want to bother, but you can see in the paper, there are also equations for the downstream processing. So you make an extraction, you have the equation to measure which is the impact of this extraction, or you work in a process in an organic solvent, you make equation to know distillation of this organic solvent, how is going to be the cost in terms of CO2. So with this approach, we tackle the full LCA for projects that we could manage, and a smaller kind of LCA rapid tool to know a bit where we are and how to improve the things in the lab for the beginning. And I think this is the end of my part. Thank you.
Would you pass me?
And then, Heiko Keller, please.
Thank you. So I'll try to run it from here. Maybe you could put up the slides. So I'm Heiko Keller, currently Senior Project Manager at IFEU, Institute for Energy and Environmental Research, Heidelberg in Germany. 20 years ago, I used to purify proteins in the lab, so I really know all the hard work that's behind what you're presenting here. Now, what I'm doing since then is sustainability assessment, starting from LCA, but also including the whole variety of impacts that should be covered or could be relevant. That is our toolbox we're applying to usually something like 10 EU projects at a time, together with my colleagues in the team. And beyond these three standard pillars, using LCA, LCC or TA, and social LCA, we think that there's more aspects to be considered if one wants to make some process from research to be a sustainable industrial process. One thing is that one should have a look at risks and barriers, also like Gro mentioned earlier. I think it's really important that what one is planning to establish can really become reality later on. And then there's also local environmental aspects which are not covered in LCA. There's social benefits, and these all these messages we try to condense into trade-offs and really tangible recommendations for the partners to go ahead. So the reddish ones here is what we have tackled in the project EnXylaScope, and the gray ones were either covered elsewhere in the project or not relevant in this case. And it's also other tools from our box. Our goal is strategic decision support to make these processes real and sustainable. But it's not only about assessing something with huge variety of indicators, it's also about thinking about how this whole process looks like from cradle to grave, to piece all these elements together, which are usually covered by highly skilled experts, but these interfaces and connections, they need to be made. So for us, the approach is to have, for example, workshop at the beginning of the project where we gather all the experts and together set up some questions of what could really help everybody to find out, so that we can set the goal and scope without a lot of technical phrasing, and to also design the scenarios together, to put all these things in a big line from these experiments to a whole scale setup that is fair to compare to existing technology. In this case, we ended up with several chemical engineering models of 15,000 ton biomass input by refineries, as the colleagues from Selignus have modeled it. And that was the input to our assessment then in the end. I think there it is important also to think beyond the pilot scale and to think beyond today, because in 2050, when these new biorefineries are still about to run, it's a totally different world from now. So just calculating with these database figures from 10 years ago, it doesn't really give you necessarily the right directions. And then it is also especially important for enzymes to also have a look at indirect effects outside of the usual scope. It could be co-benefits that cannot be really represented in the functional unit, to have these complicated LCA terms. But if you would like to know more about it, that's something we also presented at the last EU BCE conference. And parts of it is also what one could summarize as handprint, as you mentioned it before, to simply see it's not only the footprint. If you have high-value products, they also have a performance and they help save environmental and other impacts somewhere else. And with that, I would like to thank you for the opportunity to be here in the panel.
Thanks. So, Monique, in your project, in your sustainability work, where do you see that? Where did you have the biggest? If you want to pick one place during the project, where did you saw the biggest impact of your work? And was that surprising?
Yeah, I think the biggest impact was if you look in our process, then yeah, let's say we first make the molecule, the quite big molecule, and we could not change much there. We also don't want to change much there. But the biggest impact is in the next step to really cleave the molecule. And there, what was quite surprising for me is that that even we could not yet make full LCA calculations because we do not have all the numbers yet. But what the pieces what we could compare is that you see already that even that small step has such big impact on the total. So that was really really nice and surprisingly.
Yeah.
Really
you got
good.
the right result.
Yeah, exactly. Yeah.
Okay.
Yeah.
Pablo, the same question to you.
So I think one thing that we realized, we all in the project when we started to do this kind of numbers was the importance of the conditions in having good numbers in sustainability, and then people started to be more aware of the fact that they need to make intensified processes to get things in the right way in sustainability. And also, I would say one aspect that was completely overlooked at the beginning and then it was realized was the downstream processing. So people, I think now there is an awareness in the people that work in RadicalZ that this is quite an important part that needs to be addressed from the very beginning if we want to have a sustainable system and so on. So I think this is something that probably is a gain of the project. And also the identification of some kind of solvents or things that we can use or we should not use, and then start to work with the good ones from the very beginning too. So trying to incorporate this when we are still in the lab scale, I think this is quite important message or lesson that we got. And I think it is a success for the project, I would say.
Thanks. And Heiko?
I think what impressed me most during the project was to see how this whole process developed from lab experiments to this model of a full-scale biorefinery. And of course, that was rather before our core work, but we could be a facilitator to put all these elements together to some picture of a goal to go for industrialization. I think that impressed me most. But I also handed on your question to the consortium to see what the partners were interested most in. And I think it's, as in most cases, lots of details here and there. And from that, I take that the whole life cycle perspective is important. For example, to see that biomass doesn't come just in a bag with a price tag on it, but that you have to know about its origins, its history, to be able to cooperate with the farmers later. Or also insights from the real LCA calculations then in the end that, for example, the enzyme modification of the xylene helps to save environmental impacts, although there is an additional step, just because the downstream processing is easier then. So these indirect effects need to be taken into account, and I totally agree with you, the biggest progress has been made in downstream processing and the whole integration of enzymes into the process, and I think that is the biggest step ahead there I could see.
Yeah, I fully, that's also what we found out in OXIPRO, the indirect impacts should also be included in the overall calculations. Thanks, very, very nice replies. We had in OXIPRO also worked a lot with the safety and looking at environmental and human health. And that is, of course, when you look at the use of enzymes in the EU regulations, it is a question that we have been also tackling or discussing in the project. Now, Pablo, you looked at you developed this tool or calculation methods which sort of like gives like a quick and not dirty, quick and clean way of calculating the carbon footprint. How do you see? Could you sort of like have is it possible also to make a similar tool for assessing the environmental health and safety? Or is it too complicated issue?
Well, I think the first thing that I need to say here, and you are right, this is life or process impact does not end in CO2. CO2 is one of the components, but you have shown quite a lot of other things that are important. I'm thinking out loud here. I think any property that could be linked, could be related to the mass of the wastewater or solvent we are making, or could be related to the energy that we are using, probably there should be a way to link and to make an estimation method. There are other properties that probably they are or they are not, and probably those I don't think you can make, by the default if you are using a solvent, and in the MSDS it says that it is teratogenic, I'm saying something, then of course you already know, but all properties that depend on the exposure dose that you are, I guess that you can do an estimation based on an equation, which is the amount that you are using, and based on that, try to find a link to the limited dose that you can handle per time or per something. I would say that it could be possible. I think.
If I take the word here, I see also that there is then should be a difference between the occupational exposure and then what you actually find out in the end product that goes to the consumer, because they are here.
Absolutely.
The use of solvents are maybe not that relevant in the end product, but very relevant when you go to the exposure to the occupational health. So this is again, the more you know, the more complicated it gets.
Yes.
And that leads me to the next question, actually. Heiko, you had made a very large matrix of including all the 3 pillars of the LCA, the social, economic, and environmental, and also looked at different scenarios. That means that you end up with, let's say, a lot of data, a lot of information. Like, on the other hand, this is good, but then looking at from the other point of view, how do you come across with such complex, diverse set of data to your stakeholders? Do you have any quick and clean suggestions for us researchers to avoid this? But on the other hand, it's not that, even though I said it.
Well, in the first place, I wouldn't consider our results to be the numbers. I mean, these numbers are anyway just snapshots because the economy is developing so rapidly towards decarbonization and whatever else is happening. So you could update these numbers every few years and they would be different. What's more interesting for the stakeholders, I think, is recommendations and how to improve it. It's also just having 70% of CO2 reduction is great, but still there needs to be pathways to go to zero, because that's what needs to be in not too many years from now. So our results would be recommendations, and we have a full list of them in our deliverables, and that is not that all the stakeholders should read it all and connect all the dots and pieces, but just to pick out a couple of messages they can tackle, they can take to improve the upscaling and so forth. So I think the most interesting part for me is always if some of the project partners or stakeholders are then like, yes. So if there's this moment of understanding where I think we could help them to get a step ahead in the development of a process.
Thanks. So one more question, I think we have time for it, and goes to Monique. And you've been working with the cosmetics, and you also mentioned there is a huge demand from the consumer on more sustainable cosmetics. How do you see is the industry able to produce in the same speed greener or more environmental beneficial ingredients, or are there any bottlenecks there?
Yeah, I think there are a number of bottlenecks. I think especially younger people are very much interested and have a much higher awareness of, let's say, cosmetics, which are more greener or sustainable, or how you want to call it. But at the same time, they want the same performance and even better. So the real challenge for the industry is, and I think also for the science, is to come up with more greener processes, but additional with at least the same performance, even better. And then our, let's say, our customers, so that are normally the big cosmetic companies like L'Oreal, etc., they don't want to pay more for it. So it's really nearly impossible, but you want something which is better, is cheaper and greener, and that's always that is a real challenge. And I think the demand of the consumers in general is really more on the greener part and more sustainable. And I think it's our task to come up with the right answers, but it is a challenge. And I think what was nice in this project that up front I would not have thought that we would be able in just 4 years to come with this result, and that was really so nice, because it's really a combination, let's say, of the science, of upscaling, of, yeah, let's say all the different partners, but make it so efficient, and that I really like about the project. And I think we really need more like this to meet the expectations of the customers. Yeah.
I think we can round up then with this lovely message that it is impossible, but we did it in 4 years. So.
Yeah.
Thank you.
Thank you.
So thank you for all the speakers, and thank you, Mona, for moderating. We are moving to our next roundtable, the Raising Awareness. So the moderator of this roundtable is Ana Lozano. With a background in biology and science communications, she has around a decade of experience in both discussing sustainability and communicating in front of the public as an interviewee, interviewer, speaker, and moderator. So, as she is doing today. She has experience communicating in the computational enzyme engineering sector, and right now she's communicating the science that is cooked at different Spanish research centers as well. She has put aside those obligations today to be back in the enzymes arena, and does have her English to guide the conversation in this interesting roundtable. So, Anna, you may take the stage.
Thank you very much, Nerea, and good afternoon, everyone. Let me invite our guests to the stage, please. So that we can start this second roundtable. As you have all in your programs, it's going to be about exploring how communication, education, and engagement drive sustainability and how they must evolve for greater future impact. I think we're missing 2 of them. Yeah, okay, they're putting their microphones. Let me say a few words to introduce this roundtable while the rest of our guests are coming, and in a few minutes I will introduce you our guests. Because although enzymes can make industry and end products more sustainable, we all know that in this room, we need everybody outside of the room to be aware of this as well, right? As long as we want enzymes, to be part of the industry and to really expand, and to create a more sustainable world. That's what this roundtable is about. We've invited one representative from each of the 4 projects, whose role, among others, is to make these messages understandable, appealing, and trustworthy for all the stakeholders, from policymakers to investors, public institutions, and consumers as well. Our guests have been trying to raise awareness about how enzyme-based products contribute significantly to reducing the environmental footprint, thank you, of market sectors such as cosmetics, textile, food, or housecare. Products we all use, which have a big impact on the planet's health and our health as well. But there are some challenges they face when trying to communicate their messages and outcomes, which have to do with science literacy of the public. Thank you very much. Because we all know that talking about enzymes and enzyme engineering is not easy at all, but also with the trust in science, in the process of communication itself, and in green promises we are getting used to hear everywhere, which end up blurring the honest ones. Nowadays, there are plenty of channels to communicate through, and many different audiences we need to engage with. So communication strategies are key to succeed in, say, raising funding, promoting new regulations, or motivating consumers to switch to enzyme-based products in their daily lives. So let me introduce you to the 4 members of our roundtable, who will explain to us some of their strategies to raise awareness in the frame of their projects and to engage with different stakeholders, as well as the challenges and opportunities they've found. So first of all, hi, Sara Daniotti, from FuturEnzyme project. With a master's degree in advanced biotechnology, Sara is currently head of the bioeconomy project management unit at Consorzio Italbiotech, an Italian non-profit organization that brings together stakeholders in the biotechnology sector, leading scientific development and knowledge transfer. Over the past 4 years, she has contributed with her expertise in project management and communication for the FuturEnzyme project, combining these skills with technical knowledge in assessing the environmental and social impacts of innovative products and processes. Right there, we have Susan Holman representing OXIPRO. She holds a PhD in synthetic biology and has more than 20 years of experience as science manager. She is the founder and managing director of SV Science Management, a company dedicated to consulting scientists in third-party funding matters. They also conduct research in the field of responsible research and innovation with a focus on ethics, open science, and more recently, stakeholder engagement. The latter is what they are doing in the OXIPRO project, where they lead a work package focused on stakeholder identification and engagement. Right here, the second one, we have Joanna Oliveira representing EnXylaScope project. Joanna holds a master's degree in marketing and is currently one of the team leaders in the European funded projects department at LOBA, a Portuguese marketing and communication agency with a strong focus in European projects. While she is stepping in today on behalf of the EnXylaScope communication and dissemination team, Joanna has been closely involved in similar EU-funded research projects across several scientific clusters. Her experience in translating complex scientific work into engaging communication strategies offers valuable insight into how we can better connect science with society. And last but not least, Lucas Sanchez-San Pedro, representing RadicalZ project. Lucas is the co-founder and director of ScienceSeed, a science communication agency based in Madrid. He holds a PhD in molecular biology and conducted research at the Spanish National Research Council and at Yale School of Public Health before moving into science communication. As a journalist, he has contributed to several major national media outlets covering topics ranging from biomedical innovation to public science policy. At ScienceSeed, he leads communication strategies for numerous EU-funded projects, combining both creativity and scientific rigor to make research more accessible and impactful. We don't have much time for this roundtable, so let's go directly into our topic so that we can have a few questions at the end if you want. So let's start with the first round, in which I want each of our guests to give us, in around 2 minutes, an illustrative example within their communication strategies of how to engage with stakeholders and try to improve their awareness, especially related to the sustainability dimension of their projects. So if you want, please, let's start with Sara Daniotti from FuturEnzyme, sorry.
Thank you. So what I want to bring today to this roundtable is our addition that we made to communication related to social assessment, and in particular, as our project is targeting consumers' products, so textile, cosmetics, and detergents, so something that consumers use every day, we were wondering, are consumers interested in what we are doing and the innovation that we are bringing to the project? Especially the need for such a study lies on our assumptions that enzymes cannot always be perceived as concrete by the consumers, because maybe they do not have the technical knowledge. Enzymes are very small, so they cannot touch them, they cannot visualize them. And so we wanted to know if they were actually interested, or if they could actually understand what we were doing in our project. And to do that, we performed this choice experiment study, which is a research methodology that is used to evaluate the preferences of a consumer by asking them to choose between different options that are characterized by different attributes. So, for example, will you choose detergent A with this characteristic and this price, or will you choose detergent B with different features and a different price? And by asking them this type of questions several times with different features, mixing the features, you can really understand which features are more interesting for the consumers and the value that they can give to these features. So in terms of willingness to pay, so how much they would like to pay for this type of product. And this is very interesting, first of all, to guide the research, also towards something that the consumers are interested in. It is interesting to give a value to our innovation by giving them a price, but from a communication point of view, it's important also to understand if our consumers and our stakeholder understand the value behind our research. And what emerged from our first study, we are still finalizing a few of them, but what emerged from the first that we did on detergent is it's confirmed a little bit our first assumption that sometimes enzymes are not perceived as a value. Because, and what we need to do is really to change our communication also to be able to make these invisible molecules, these invisible enzymes, to make them more concrete. So my takeaway message is that it's really interesting to try to combine these assessments in communication activity, not only to guide the research, but to guide the communication activity itself and make it more effective towards our direct stakeholders, in this case, the consumers.
Mm-hmm. Thank you very much. Yeah, exactly. Starting a conversation, let's say, with this service, with the end consumers, is a good way of knowing how they are perceiving these products, right? Let's ask Susan Holman to make her intervention from OXIPRO project.
Yeah, thank you very much. So from our experience, our strategy was a bit different, because we thought it is a good idea to include feedback towards the researcher, that they can take up the feedback from the consumers for further applications and further development. So co-creation was a keyword. It was also an invitation to the researchers to leave their comfort zone and to think about what happens afterwards. So research is never a dead end. Knowing what happens afterwards is important, and also knowing that maybe a product is not necessarily needed on the market because we already have so many products. And so we also implemented events like innovation boot camps with the youngsters, which was very nice because some of them are here, Laura and Ruben from FuturEnzymes, I guess, so from a sister project, joined our innovation bootcamp, and we did a street survey and a mini market analysis, and we all were shocked how many detergents are already on the market consisting or containing enzymes or sunscreens, and finding the niche and make it sexy for the consumer, that our research is really useful. This is a challenge, but here we need the feedback to the researchers, that they do not focus on finding an enzyme, how to use this enzyme to be successful. This is the challenge we faced, and we try to implement this also in the project and in our policy briefs and the activities. So this is really a two-dimensional conversation. So it's not one direction, only information provided. This was done by Leslie from the dissemination work package, but it was more a bi-directional conversation, feedback, question, feedback, and yeah, still ongoing. So I just invite everybody who did not take part in our survey. I will distribute this. This is very important for us to learn what you think. It was also an experience how to set up a survey. Also, our researchers are stakeholders, and this is something people are still not aware, that everybody is a stakeholder. It's a question of the perspective, right? So this is what we tried, and I think at the end it was very successful. Now it would be wonderful if we could proceed another 2 years, but we will definitely go on with our information flow and share the results.
Mm-hmm. It's a never-ending conversation, right? And it's very interesting, this co-creation process after these conversations, no, with the consumers and with the future consumers.
Definitely.
Very interesting. It's time now for Joana Oliveira.
So hi everyone, it's really nice to be here and really nice to be in this table. For EnXylaScope, one thing that we wanted to do very clearly in the beginning was to strategize the best way to communicate to different target groups and to somehow find a way to bridge this communication between all audiences. After realizing that it's really hard to translate scientific jargon to very easily and plain knowledge to the general audience, for consumers, for other people in the audience, we realized that we kind of needed a way to convey our messages that we could do for everyone. So not just the scientific part, not just the academia, not just the general public and the consumers, but everyone. So we decided that one of our best tools were videos. And with this in mind, throughout all the lifespan of the project, we developed more than 20 videos in several formats. So in interviews, in presentations, informative formats, and now we're actually in the final stages of developing a very interesting documentary style video that we filmed directly with our scientists in the Salinigs lab, where the scientists actually showed all the process behind the technology and the innovation. And I believe that bringing this human touch, bringing the human side behind the science helps the audience feel that the science is more real. It's more tangible, it's grounded, and people will relate better with this knowledge than just seeing graphics and numbers on a very long spreadsheets. So we found that developing these very high professional and quality videos helped us reach all types of audience and actually keep the audience engaged throughout the different lifespan of the project. As you all know, I believe all my colleagues know, how challenging it can be to be in 4 or 3 years projects to keep the audience always engaged. Especially in the beginning, because we don't have a lot of results to share, so we kind of have to develop a lot of different techniques to keep them engaged and knowing more and keeping coming back to us. And we found out these videos formats were really, really interesting, especially the interviews with our partners. So we were really, really annoying with them and asked them several times and brought our filming crew everywhere we went, and we developed more than 20 interview videos, and they are now hosted in our YouTube channel. That is, all the videos have more than 9,000 views, so we can clearly see that the audience is actually interested in this, and they want to have this dialogue and this communication, so that for us is quite a lot of success, I must say.
Thank you, Joanna. With the first two interventions, we've seen how the public perceives our messages, but with Joanna, we've opened the other part of the communication process, no? How to engage, how to make those messages understandable. Yep. We know it's not easy in your projects to make those messages understandable to the whole public. And I think Lucas is going to be talking about something like this as well, right?
Yeah, I think, well, hi everyone. And the question that was complicated because in a European project you have to do so many things in communication for many different audiences, so selecting one thing is kind of complicated, but to complement the panel, I think that in our case, if we have to highlight something in the RadicalZ project, is that we wanted to do something special for the kids, for the children, for the future consumers. So we know that for teachers it's different in different countries, but it's kind of difficult to develop high quality communication products for them. And we wanted to also to share all the thrill that you get in the laboratory with the discovery of new enzymes to them. And we find like storytelling is the best way to do it. And we wanted to combine it in two formats. One was a comic for kids 8 to 12. And another thing was a digital escape room where they can, like have also that frustration of not discovering enzymes in their way. So we developed these two things, the comic and the escape room. And we had the opportunity not only to develop them, but to test them before and to control that the products were actually good and functioning, and then share with the schools and see the results. And the results are quite amazing. We have like hundreds of readers and users, and it's amazing, like having the possibility of being in a European project where you have the funding to do these things at a high level and share them freely to everyone. I think that that's one of the things that are key in our case.
Exactly, it serves to everyone, right? It's not just engaging with future consumers. It's, I think, making a good progress on our society scientific literacy as well, right?
Yeah.
With these products that everybody can access and that really explain in simple words something very, very complex, right? With this first round, as you have seen, they have already used one word, all of them, which is challenge. I think all of you have mentioned it, and that's precisely what I wanted to ask you about right now for this second round. I would like you, before giving you the opportunity to make some questions if you want, in this case, I want to invite you to describe the main challenges, or one challenge at least, you have faced when implementing those communication strategies. And if you want, in the same intervention, I would like also to know the opportunities you have found communicating the outcomes of these projects to raise awareness about the importance of enzyme-based products for sustainability. So if you want, we can start the other way around with Lucas first and then continue.
So, I mean, I think that the first challenge is the one that we all share, that we are sharing raw scientific information that we have to translate. So, and for kids in our case was truly complicated. Then contacting all the schools to trying to synchronize and see what the opportunities during the year that were like easier for them. Then you have the technical challenges of programming and so on. But I think that I want to stress that one of the challenges that we normally face in European projects is that sometimes researchers or the coordinators of the projects do not understand what we do and why we do it, and that wasn't the case in the RadicalZ. So I want to thank Aurelio and the whole consortium. We translated the comic for 11 languages because they help us and they wanted to collaborate in the project. They saw that this was like two nice activities that weren't in the grant proposal at the beginning. So I think that having that flexibility and that support is super important in our projects.
Exactly, creating more products than you were expecting and which are very, very useful for all the public.
Sure.
Thank you very much. Let's continue with Joanna.
So just to share something different than Lucas, that was one of our challenges as well. I think for these very technical projects, one of our main worries was to not exactly how to communicate, but what can we communicate without breaking or jeopardizing any IPR thingies, you know. So confidential development can be a little bit intimidating, especially for someone that is not from the technical world. And we just want to communicate and we just want to raise awareness and we want to communicate a lot of things. And sometimes we need our scientific partners to say, well, wait a little bit. Okay, we cannot share that right now, or we cannot share that in that way. So for me, I think that is one of our biggest challenges, but also an opportunity, because you have to think outside the box, because you cannot share everything. You are almost obliged to find different ways to communicate and think outside the box. One thing that is for me also really important when communicating projects is the ability of communicating within clusters. So I think this is a privilege, because when you're communicating to a very wide range of audiences, not just my project, but all the projects, we're reaching such a bigger community, and we're also keeping them engaged throughout all the lifespan of the project. Because even if my project doesn't have a lot of results or preliminary results right away, maybe other projects from the cluster have, and we can communicate it and get our audience interested in their audience and their project and achievements, and we do this through all the projects. So I think a big opportunity is communicating within the cluster.
Important for your daily work.
Yes.
For these 4 years of success as well.
Yes.
Yeah. Very good point. Let's continue with Susan, please.
So in our case, it was the biggest challenge to face the GDPR issues because we needed to go for interviews, for questionnaires and surveys, and we had really to take care that the individuals are safe. And implementing tools, identification of tools, and get in contact with people, always asking for informal consensus felt years. So this was incredible workflow, more than expected. Another thing is getting in contact, especially with stakeholders from policy. We needed to write hundreds of emails and repeating and bothering them. The same applies for other stakeholder groups, but this is really a challenge and takes really, a lot more of effort than expected, because you cannot write a general email, you have to get in personal contact with them. But the opportunity, if you are in contact, that's another dimension. And if you are in contact, the communication is working very well, and we have good feedback, and we have earned a lot of insights and learned also a lot of perspective from the other side of the coin, so to say. So this was a big opportunity, but the efforts to be done to be compliant with data privacy and GDPR, especially across 13 countries, is really dramatic. So here it would be nice to have a bit less bureaucracy, let's say.
That's a good also take-home message from that point. And Sarah.
Thank you. I don't want to repeat what my colleague says, but I agree with all the challenges that you mentioned, and also it was interesting to know the opportunities you found on that. What I want to, maybe I'll start with the opportunity that there were in our project, because we have this privilege to have the free companies really representing our, always very easy to communicate, so we had to find some ways to make the communication more concrete. So as Lucas said, trying to, but also Joanna was saying, trying to make communication visual, as most visual as possible, with videos. We had the comics, and we had these very nice original images for the project that we're trying with comics or graphics to really explain what the project we're doing. And another opportunity from this challenge was also the fact that we were able to involve as an advisory board one expert in for a consumer association. So she helped us also to shape the communication toward consumers, to hold a few webinars that were able to collect a different a good participation from consumers itself. So this was also an opportunity that we have in our project.
Well, I think we've covered the whole communication process with all your interventions. We've talking about consumers, but also policy, marketing strategies, and I don't know. You haven't had the opportunity yet to interact among each other and also with the public. So I think that for a few minutes, if you have any questions or if you want to address any of the topics that the other guests have addressed, it's the time. So we open the conversation, yeah. Gracias.
So thanks. So just a comment, something happened. It's not a question, it's just to share something. When we organized the event, I sent a lot of email, to the government, the scientific advisory board of the member of the government, the Ministry of Ecological Transition, to local, to the FCC. All the people reply almost immediately. Well, I cannot, but you contact. And then I sent to the consumer organization, a Spanish one, no reply at all. Even I told that in FuturEnzyme we are working with Ultra Consumer, which is a really well positioned. So I think somehow I'm missing something. That even if the consumer organization in Spain, which is really well recognized, the OCU, the OCU, really well recognized, and others, so it was the only stakeholder that didn't reply to me. And I was impressed because it is consumer product. And then, I mean, I'm not asking, but I think still we need to convince even the general consumer organization, because they are they have the priority position to convince the consumer what we are doing. It's just I want to share this.
Does anyone want to reply?
Yeah. So we made the experience that you have to repeat the writing, repeating, because people are overwhelmed with requests and requests for filling in surveys or something in this direction, and we made this experience, and this is painful because you put so much effort in and nobody is answering. So this is really not nice. But we also learned, and this was in the beginning when we set up the first questionnaire, we included the researchers for development of questions, and they expected written answers, and this was too complex. So then we had to simplify the questions for yes or no answers, and that works. Yeah. So this is really something. But anyway, you have to really write emails, I don't know, thousands of emails to get 10% answers. So this is unfortunately the case, but I think each of you receive so many emails per day, and probably the requests for filling in surveys or will you participate something go directly to the waste. So that's our life, unfortunately.
Well, there's time for one more question. Yeah.
Nice to hear about the complementarity between the projects. Very nice. But now, in retrospect, projects are coming to an end, and you've had your challenges. Do you see other opportunities, other strategies you would have chosen instead? Now, looking back, could you share some new ideas with us?
I would say just a comment that the same comment that you mentioned as coordinators of the projects, that we have a lot of fragmentation. At the end of a project, you have created a community after a lot of work, and then we have collaborated. We could collaborate more always, and this is something that the Commission wants, and it's super useful. But at the end, we need a hub. We need something that at the end, take all the legacy of the communication, all those communities that are already engaged. They are going to die if we don't feed them in the next years. And when the projects are over, there are some gaps. We can move some efforts in projects that we are starting now, but at the end you always lose people in the way. So
I do agree with you, Lucas, and I think, and I've seen this in other projects, perhaps it would be a really good thing to think forward. In other projects, in my past experience, especially in clusters, we have created one unique platform where all the project results and achievements were placed in it. So instead of keeping alive 1, 2, 3, 4 websites, instead of having other platforms and social media, we collected everything in one. So it was easier to keep it alive. We for sure know that with the time passing, and other projects also researching similar topics and evolving, so that research will kind of die a little bit. So I think for future proposals or stuff, having the clear thought of implementing a universal platform for clusters may be a good idea.
Well, thank you very much to our guests for all your meaningful interventions. We are out of time, so thank you very much and congratulations for these 4 years. Thank you.
No. Thank you, Anna. Thanks to all the speakers. So we are now introducing our last roundtable about industry implementation. And for it, I'm welcoming to the stage Tobias Clement. Tobias Clement is a biochemical engineer by education. He has studied at RWTH Aachen University in Germany and worked at the Center for Molecular Transformation for some years after his PhD to coordinate large joint projects between industry and academia. Since 2018, he's Deputy Cluster Manager at CLIB, the Cluster for Industrial Biotechnology, where he is responsible for strategic development and interdisciplinary consortia. Welcome, Tobias.
Thank you. So I will try to buy the technicians some time and briefly introduce CLIB. We already heard it's a cluster of industrial biotechnology. So CLIB is an association of 120 members, an open innovation international cluster, and we try to connect our members. And our members are from large industry, but they are also from smaller companies, startup companies, research institutions and universities, as well as from infrastructure providers. And our members, I think they are connected. They think biotechnology is one key tool for the challenges that are ahead of our society and also of our industry. And yeah, let's phrase it that way. We are trying as CLIB to bring those people together, to connect them, to create interfaces between our members and beyond. And we think, yeah, international consortia are a great opportunity for this. And of course, it's always interesting to see how such consortia develop. What is about, yeah, you start and at some point you want also to see some kind of impact. And I think now we'll talk about what is this impact and yeah, how we can accelerate impact in the projects. I would like my participants to come on stage. So let's first start with Laura and Xenia and Nicola and Christian. So, Laura is from Kerry, and she's working there as R&D&A manager in the enzyme department and is responsible for human nutrition, but also for pet and animal nutrition. I think you have described yourself as enthusiastic agriculture engineer in LinkedIn.
Yeah, you were stalking me, right? I stalked you before.
Yeah, and the next one is Xenia. Xenia is a bioscientific fellow leading activities on enzyme discoveries for chemical synthesis. Then we have Nicola. He's the co-founder and CEO of a startup company of Gecko. And last but not least, we have Christian. Christian is working for the company that is now called Novonesis. It was former, I think, Christian Hansen for you.
I come from the Christian Hansen part.
Yeah.
Yes.
And he's also working on enzyme development for dairy applications. Yeah, perhaps you can introduce yourself a little bit more, tell something what you're doing at your companies, but also what was your work in the 4 products for greener products.
Thank you. Yeah, so as Tobias introduced, I'm R&D manager in Kerry. I've been working in the application area. So within Kerry, we have like the fermentation team and the application team. So we're working, once we have new molecules and new enzymes that we want to try in new markets, we do all the application work to see how the enzymes work. So it's very important to find innovation that we could develop new products and they are greener. So some of the things that we focus in Kerry, because sustainability is one of the big pillars in Kerry, is like how we can reduce carbon emissions, how we can produce increased yield, and how can we reduce food waste and optimize local resources, especially, for example, in animal nutrition, we want to optimize like crops that could we use to optimize digestibility in animals, and similarly in pet and the other areas. Yeah.
Yeah, please go on.
Oh, you can hear it? No? I don't know.
So my name is Ksenia Nizel, and I'm working at Bayer. And within Bayer, we have biotransformation platform where we're developing enzymatic solution for chemical synthesis. So it means that we start from the identification of the enzymes, I mean in microliter scale, not in your picoliter scale, and until the 100, 150 cubic meters. So we have actually all the activities starting from the discovery, high throughput screening, protein engineering, fermentation, downstream, and so on. And within the last 4 years, I had a very great pleasure to work as advisory member, as a member of advisory board in FuturEnzyme, and I'm really happy to see how the project was developed, and also enjoying the fantastic atmosphere and a collaborative spirit of the project, what was really very amazing to see.
Hi, well, mine works. Sorry. Hi, my name is Nicola. Well, a typical answer, what Gecko is a startup company that supports enzymatic development for companies like Novo, Bayer, Kerry, and similar. Sometimes also companies that have no experience in using enzymes, but we also like very much working with companies that do have experience in working with enzymes, because the language that we speak is the same and it's much easier to work on that. And I noticed also in the previous session that there are many challenges, and within our consortiums we have challenges because we speak different languages in science and market research and regulation. So that would be my typical answer, but I think what we are actually doing at Gecko is we are learning, and that's a continuous process, and it's painful because there's like so many different things that we have to cover. When 4 years ago, when OXIPRO started, we were, I think, 4 people, and now we are 16. And Gecko is, well, company is growing organically, so it takes a lot of effort of all the people that come in to do so. So I'm happy to participate in these kind of projects and to build capabilities through them. Thank you.
Yes. Is my mic working? Perfect. Yes, my name is Christian.
No, it's not.
No? It's not?
Yeah, it is. Okay. Sorry.
Okay. Everyone agrees it is?
Perhaps we can have a public vote.
Double.
I shouldn't use 2 mics then. I think that's going to be not
Yeah, that's
for
why.
this reason. Yeah, good. My name is Christian. I'm representing Novonesis. And as you mentioned, it was when we entered the RadicalZ project 4 years ago as Christian Hansen industrial partner. So Christian Hansen is, or used to be, basically a global player in ingredients, biological ingredients, so microbial cultures, enzymes for dairy products mainly, but also for agriculture. And then so we are located north of Copenhagen in Denmark. And about 1 year ago, we merged with our geographical neighbor, Novozymes, to form Novonesis. And now we basically deliver biological solutions for a very broad range of industries, household care, the major like of Novozymes, as you might know, bioenergy, carbon capture, food, health, and so on, right? So you could say, if you talk about sustainability, that's in the DNA of our company. So, and within the project, we were working on the development of enzyme products, discovery of enzymes for dairy products that are supposed to work in situ in dairy. So they work directly in the product to generate healthy molecules, so for consumer health. And as well in developing technology to deliver enzymes within the dairy product to the place where they're supposed to act. That means that the process, the manufacturing process for the dairy plant would be faster, more efficient, and therefore more sustainable. So these are the two legs where we are contributing with to the project.
Yeah, that's great to hear. So we're now nearing the end for those 4 projects. And perhaps you can elaborate a little bit more. Was it easy for you to work together in the projects? Have there been some hurdles? How do you overcome them? And just working together with those academic partners, you're all from industries, or let's say on the other side of the Valley of Death, the infamous Valley of Death. So, perhaps we could start the other way around, Christian.
Yes, hurdles. I mean, hurdles are there to be overcome, right? Of course, there are learnings. There are aspects maybe to be aware of before going into the project. So it's a bit, I see the upside and the benefits going together with also the learnings, or I wouldn't call it downsides. Mm-hmm. There's always 2 sides, right? So maybe I start with what I think was the major benefit for us, or for me as a researcher or development engineer in industry, is that you get, with such a project, you get the opportunity to do explorative research, to really to say, okay, we test hopefully disruptive, but novel concepts, novel technologies that often you don't have the chance in industry. Maybe you can start it up with totally new ideas, but you will always run into some difficulties, and then often you lose the focus, or you don't have the chance to follow up on it. So here you have an agreement, a funding agreement, you have a collaboration agreement, so you continue. So, but this also comes with the downside that you have this agreement, you have a long-term agreement and a project plan, which is not so common to have in industry, right? So in industry, basically you can change direction in a project, you can stop a project within a day, literally. So if all the stakeholders agree, or the important stakeholders say, we will never reach our goal, we should stop here, right? Mm-hmm. So you cannot do it in such a project, which then in turn tells you, or the learning is, you shouldn't be too ambitious in going in the project, which is a pity also, because you want to be ambitious, you want to come up with disruptive, as we call it, right? New ideas that really do something great on the market. But you need to be careful, because you cannot just stop it and say, I do something totally different, because you have collaborators and you have an agreement. That I would say is one of the difficulties or the learnings.
Yeah. Nicola, you're a small company, you said you have grown a lot in the last 4 years. So it's also kind of a journey for your company, not only inside the project, but also outside. Would you also agree that this is a challenge?
Yes, I think in this kind of projects where we have also a range of different partners, from excellent academic institutions to, well, very large companies. In our project, Novozymes at that time, and now Novonesis is also a partner. We also had Rolco as a detergency company, and understanding that how difficult or unusual challenge can be, same as Manuel showed this text that has to be treated in 30 seconds. And then for half of the people on the team, the instinct is like, this cannot be done. But then for the other half, it's like, okay, if we are going to do this, what do we need? And we do have 4 years to try to do it. I think it was quite productive. We have reached certain stage in development. We have filed a patent application on something that we think it will be valuable. We also learn how slow is the process and how late we will find out if it's really going to work or not. But the collaboration within these consortiums is a way to solve market problems, because individually we cannot. And you can see very often that in academia there is a lot of brain power and, well, puppeting hands trying to solve something that also very often they think it's a solution, but once you cross on the other side where we are now, and after many negotiations and NDAs and other things, you realize that the answer is it is not interesting. Mm-hmm. But you don't have access to this knowledge as long as there is no direct collaboration. And very often there are reasons to govern not to collaborate on this, like competition.
Mm-hmm. Yeah, Laura, I thought you're agreeing a lot. You say,
especially with Christian, because coming from business and from industry, what I have seen is like the change on direction in some projects is like if something is now tangible and you don't see results or you don't see money coming from it, you kind of stop working on what you were working for some years immediately. And what I have seen, especially working in the EnXylaScope project, that obviously at this point for Kerry, it's good to participate, being involved with different partners in a consortium, but it's like difficult to justify the resources to like progress in the project. And another hurdle that I have seen in this project is like working in one of the latest work packages, which in my case was work package 5, which is using the enzymes that we found as the best candidates. When any other partner in the project had any delay, it was also causing delays for us to progress and develop data and produce data to justify the investment and also to prove that the enzymes are working in the application that we want to work in. So that was one of the biggest things.
What did you like about the project? What worked out very well?
Yeah, I mean, still for us, we have to do some more work to fully validate the results. And it would have been beneficial for us is to have like food or feed grade enzymes to be able to do some like application trials in finished product. Because at this point, the enzymes in the project were not food grade, so we couldn't do baking trials or we couldn't do animal trials to actually prove that the results from in vitro are actually working in a real scenario.
Xenia, we have talked in the coffee break about biotechnology in a chemical company, and I think it's a very interesting perspective because we are talking about enzymes as a very useful tool for companies to become more sustainable, to produce greener product. Perhaps you can elaborate on that a little bit, how biotechnology or how the view on biotechnology has changed over the last years.
I think it comes together with the difficulties in the view of the society on the chemical industry, and that the decision that the chemistry must be greener and develop a lot of biotechnological solutions and so on. This is something where we realize the power of the enzymes and the power of the biological solutions. And also, this is something what I experienced in this project over the last 4 years, how quick the results were coming. I was overwhelmed about the scientific ideas and spirits and collaborations between universities and small companies and bigger companies. And that was really a lot of movement, and this is something what I see within our company as well. So that there is a change, there is a big change towards sustainable solutions, biotechnological solutions, and so on. So we are going all together in the same direction, and it's really enjoyable.
Nicola, obviously you believe in the power of enzymes, otherwise you would not have founded your company, but perhaps you can also comment on that.
I think a common misconception also in beginning of this field is that we can replace most of the processes with enzymes, which is absolutely not true. And it's very difficult to compete because in the end, competition is at a cost. Even if something is green, it has to be at the same cost or more beneficial. In the first few years, we were working exclusively on enzymes and enzymatic reactions that, well, they have advantage over traditional chemical processes. But since last year, we also have a few people on the team that are working on chemoenzymatic processes because, well, the combination of both solves more problems. And I think it is also more realistic to have that approach.
Christian, we already heard about the time scale. The 4-year project may not be enough. Can you tell us a little bit how long does it take from an idea to create a new enzymatic process until you could sell your customers the final product?
A long time. It depends a lot. So there is, of course, the sort of enzyme development or discovery part, which normally, depending on the challenge, of course, you can rather oversee how long time it takes. Even if it takes years, it's surprising maybe to hear that that's just the smallest part. So you have further down, you have the product development part. So you have a lead enzyme. You need to turn that into a product where you need to make a production strain. You need to get production facilities done. You need to do all the regulatory part, right? You need to get it basically approved in all the countries that are relevant. Not only the countries where you want to sell it to, but also the countries where your customers want to sell their consumer products to. And I would say so the regulatory part takes the longest time.
Yes.
At least for food, I can say. It can. It varies a lot. It always depends also on some time frames the authorities give to you, right? There might be deadlines, then there might be some years of gaps where you cannot apply. So it can go up to 7 years. I would say easily, but it's always. You always have that in mind before you go into developing an enzyme, because it's not only the technical and scientific feasibility that defines the strategy or the IP landscape, but it can be what makes the regulatory part easier. So should we engineer an existing product? So should we find a new wild type or enzyme, or should it be an engineered one? Or is it important that we can use our production strain we use for another enzyme? It's these considerations are important to shorten the timeline to the market. And the longer the perspective is, or the timeline to the market, the more insecure is your business case. So it's nice if we have a good strategy to come to the market within 7 years, but will there be a relevance for this enzyme in 7 years? Yeah. So it was a long answer to basically, it takes a long time.
I was totally aware. As soon as the topic becomes regulation, I think every one of you could talk about 1 hour about regulation and regulatory issues, and but perhaps there's also learning from that. So we heard about, for example, RadicalZ. It was about accelerating enzyme development, and so, but there's only the first phase, as you said. Then the regulatory issues come later on. So we have to keep that in mind, Laura, from the beginning on.
In the project, we were discussing with some of the partners of the consortium that we need another EnXylaScope to move to the next step, to take into account all the regulatory aspects and to be close to commercialization, because at this point it's quite far, and as it has been mentioned, it can be years, especially when it's a novel enzyme, a novel molecule. There is a lot of requirements to compile for especially for Europe. Then we have like the EFSA that we need to submit a dossier, but if you want to play in different regions, you have different associations, different requirements to be able to commercialize in different countries.
Yeah. I think our session is about reality and research. So is there a difference between reality and research? Is research outside reality, or is there still a gap, or has it become closed already? Do we need more projects like this to bridge it? Perhaps, Xenia, you can comment on that.
I do think that we need more projects like this, because for us it's really important to see what's going on all over the world, what the technologies are developing, and to realize how we can make our products quicker, sustainable, more effective, and so on. So I would really like to see more projects and more consortium growing in this field. In terms of the universities or smaller companies and the bigger companies, we have to work all together to make sustainable future for the next generation. And I guess it's really important.
Nicola, for you as a small company, I think this is something you also like about projects, that you get in contact, or is it more about the development of your product, or is it more getting new contacts?
All of it, actually. So we operate very early at enzyme development or development of processes, but through all of these projects like these, but also through our contract research projects that we do with our clients, we learn how important it is to early on account for the intended use. So enzyme development is completely different if we are talking about a bulk chemical or a different sector, if it's agri-food or if it's high-value compounds, because then you can allow different kind of complexities of the process, for example. But we do need more of these projects. I think that's for all the reasons that were already mentioned. From perspective of small companies, we do need more of these projects because for us, at least, this is a way to build capabilities. So we have a part of the team that works on projects that are ready for let's say, commercial service and development. And within these kind of projects is where we have flexibility and a playground in a way to test all the new things and new technologies to face the reality, sooner rather than later, and to adapt.
Christian, is there some way we can, let's say, concept or work out a concept for new projects to bridge this gap even better?
First of all, I would like to say, yes, there is a gap between principal research, academic research, or fundamental research, as we would call it, and reality, which is market, consumer market. But I think it should be there. Because you should not, in my point of view, restrict academic research always to will that be applicable on the market? Because, I mean, if you had this mindset completely, then I think most of the really groundbreaking inventions or scientific breakthroughs would not have happened. Where you should have the freedom, I think, in academia. So, and I think, therefore, it's important to have these kind of projects to bridge basic research and to market applicability. I hope it was answering you ask what we should do differently to set up this project, or
is there something? But I would agree it's important also to have this kind of fundamental research that is exploring crazy new things. Yeah, because otherwise everything would be streamlined and some of the greatest breakthrough inventions would not have happened in the last years. But for a project, so it is good to have both sides, but we always need a kind of connection. So the project should be aware of their...
Yeah, but I think within these projects, both sides learn from each other, right? So, I mean, how is basic research done? I mean, most of the R&D people in industry, they know at least how it was done a few years ago, because they come from that. So, but also the other way around, right? So how is this research you might do in a research institute? So what is the relevance? How would this needs to be translated, or what other aspects need to be taken care of if it transfers to industry? So these are really important learnings, but I think that's what these projects are for.
Yeah. I think, Laura, you have to catch... You have to catch your plane? Okay.
I need to go.
So perhaps, but I think that then we can come to the conclusion. Perhaps you can say one point what you have learned from the project and one wish you would have for the next round of projects, because we now hear it in the next years it's about green competitiveness. So what do you wish for this?
For this project?
For the next project. What did you learn from the last project, and what do you wish for the next project?
I mean, what I learned is it's very important the connections that you make with the different partners, because it helps making and progressing the project. That's really crucial, I think. Being able to have these general assemblies every 6 months to get everyone updates for what each of the partners and work packages are doing. And I think that has been really good and beneficial. Learning, you know, when it's in science, you always have to anticipate that whatever can go wrong will go wrong in the lab and could happen. So you need to have like a kind of contingency plan to overcome some of the issues. And what I would expect for the next project, if it's something that is continuation, is thinking of having some as I said before, food grade enzymes that you can actually use in a real application with more complex substrates. Yeah.
For me, it was really the work what our communication team does. I was really impressed about the quality of all the illustrations, what was developed during the project, because I do think that we have to popularize the science to the society and to explain what we are doing, because we are developing amazing technologies when we have amazing results, and we should be able to transfer to the society that to motivate them and to have more appreciation and recognition of the science. And for the next project, I think, Manolo, with respect of the speed of the current project, we have to think about the commercialization of the results a little bit earlier. And to think about the IP rights and commercialization, and maybe also what we are doing in the industry, thinking back. So if we are going to commercialize the product in 2030, what needs to be done, by whom and by when? And this is something what is my learning for the next project?
Yeah,
Nicola.
well, I can only agree with communication part, because, well, in the end we also have responsibility to the public that funds this kind of research, and it also brings clarity in the work. So what are we actually doing, and where is it going to be used in these cases, because these were consumer cases. For the next round, well, either to have all the goals set, or maybe the TRL starting point higher so we can reach it in 4 years, like some further steps, or to have longer projects, because our experience, well, what we can observe for many of these projects is they finish and it feels a bit abrupt, because many of these things that are left unused. And Manolo also had a lot of sequences, like what to do with them now. It's only 4 years and it's still there. So valorization plan, maybe more detailed than early on, agreed. Yeah.
Yes, I think I liked a lot in the project that it was such a broad scope. So it was, of course, the fundamental research, it was the translation to industry. I mean, that was set by the partners, right? But also looking really into sustainability, right? So making these life cycle analyses, looking into what is a true number, not just saying it's an enzyme, so it's green, so we are great, right? So really questioning, is that true? And also, as you mentioned, right, the communication, so reaching out to not the immediate stakeholders. So we talk about enzymes are the future because it's more sustainable. So how can we teach the kids already, or the young people? So how can they understand what we are talking about without drawing chemical structures and having nice microfluidics videos or structures of enzymes? You say, what the hell? So I really like that. And this was all implemented basically in one big project. For the future, I think I would take a lot of learnings for myself into the next kind of project. So it's a lot also what was mentioned, basically aligning ambitions with a lot of aspects that are with it's company internal stakeholders versus the project stakeholders, right? Which are the collaborators, the peers, the direct peers, but also the coordinator of the project, and also dealing with the direct partners, because sometimes they have different interests, right? They need also to follow. So besides you could say, normal day collaboration. So publication versus IP and this. So there's a lot of small aspects I would consider.
Okay, with that, I would like to thank you again for participating in our last discussion round for today. And now I think we'll hand over back to you.
Thank you. And thank you, Tobias, for moderating. So before I let Manolo with the final remarks for the event, I just want to say to all speakers, moderators, and participants in the roundtable, we have a small gift for you. So if you can stop by before leaving the event, just to thank you for your contribution and your participation in the event. And that will be all. Thank you very much also from my side. I will leave the final remarks to Manuel.
Yeah. So thanks, Nerea, for moderating all the morning. Yeah, and I think I'm not the person that needs to be alone here. There's Gro, Aurelio, and Carolina and others, so they participate on that. So I'm really thank you all of you for being here. I think it has been a great experience. This is the end of the event, but I don't know how to say. It's an open window for future collaboration. I think that this event is not only to demonstrate the collaborative effort that this project have done and the people involved, which is crucial. It's not the project itself, but the people that make the thing happen. But I think this is the closing of the event, but it's an open window for collaboration. We know each other, and I think we are all together involved in perhaps future proposal, as Mafalda already mentioned. So there is a new action plan. The Cluster 6 will give us for sure the possibility to collaborate with many companies, many of you. So and then I will be happy if all of you we are in contact with mails and so on. So perhaps this is the starting point for new collaboration. And I wish you all the best for the future. And thanks for all of you to participate here and then to collaborate. Yeah, and then that's all. Yeah, thanks. Yeah, and of course, thank you again for all the people involved in the Fundación Telefónica. That's really great. They have made a great job to organize everything, and then that's all. I didn't want to finish without telling this word. Yeah, thanks.